All right. Well, I guess we are right about 9 o'clock, so we'll kind of get going. Maybe I'll let all of our different speakers start introducing themselves as we kind of let everybody else get in, and then we'll get started for the morning. So if you've not met me before, my name is Madalyn Shires, and I am the SDSU State Plant Pathology Extension Specialist. I also serve as the director of the diagnostic clinic here on campus. So I'll just kind of call out our presenters by name and then let them introduce themselves. So we'll start with Marty Chilvers.
Good morning, everybody. Martin Chilvers, Michigan State University field crop pathologist. We've been dealing with tar spot since, was it 2016? Seems like a long time ago now. But anyway, it's been in Michigan for quite a long time now.
Thank you. Next one I see is Jose.
Morning, everybody. I'm Jose Solorzano. I'm a PhD candidate in the University of Minnesota, and I work with Dr. Dean Malvick. And good morning.
All right. Next presenter I see is Tamra Jackson-Ziems.
Good morning, Tamra Jackson-Ziems. I'm at the University of Nebraska, and I'm the extension corn pathologist statewide here in Nebraska. And we've had tar spot since 2021, so still relatively new to us, and it's made its way about halfway across our state, just south of you.
Next presenter, Dean Malvick.
Good morning, everyone. I'm Dean Malvick, plant pathologist and with extension and research at the University of Minnesota, based in St. Paul.
All right, next presenter, Sierra Kataska.
Morning, everyone. I am Sierra Kataska. I am a graduate research assistant getting my master's at South Dakota State, working under Madalyn Shires on tar spot and other corn diseases.
Next presenter, Darcy Telenko.
Good morning, Darcy Telenko, extension plant pathologist at Purdue University. So we've been working with tar spots since we had the epidemic in 2018.
And our final presenter is Connie Strunk.
Good morning, Connie Strunk, plant pathology field specialist with SDSU Extension, and I'm housed, for those that are in South Dakota, in Sioux Falls.
Okay, well, everyone, that is your presentation group for the day. We will go ahead and get going here. So if you're in South Dakota, we first confirmed tar spot in October of 2022 in Clay County. And it has since spread to 46 counties as of last year. And we will let Sierra kind of talk to you a little bit more in depth with that and get our program going for the morning. Please let us know as Sierra is getting ready to share her slides. Please let us know in the chat if you have questions. But we also have the panelists talk at the end of all presentations. So you've got plenty of time to think about questions and make sure that we get those answered at the end of the day. All right, and with that, Sierra, I'll let you get going.
Perfect. So I'll just kind of go over a quick update of South Dakota's tar spot 2024, a little bit more in depth than what Madalyn just covered, and a little bit about some of the research that I've been doing for tar spot across the Great Plains. So as Madalyn mentioned, the first time we saw tar spot was in 2022. It was only reported in 6 counties. Tar spot is known to be caused by Phylochoromatus, and a little bit of what I've been working on, I've been doing a genetic survey of the population of different isolates of tar spot across the Great Plains. So that includes Kansas, Nebraska, Minnesota, South Dakota, and hopefully North Dakota in the future. I included in here just kind of a visualization of how far tar spot has spread in the last 4 years across not only the Great Plains, but the Midwest as a whole, and even out into the Eastern coast. So a little bit about what I've been working on with my genetics. I have collected about 200 samples from 6 different states from the last 4 years. All of those were DNA extracted, and eventually we quantified that DNA that's in those samples, and then we were able to identify that it is in fact Phylochoromatus. And kind of what I was looking for, the main thing is looking for a single nucleotide polymorphism, basically looking to see if the genetic code of each isolate of tar spot is any different from, something in the past or another state. So in total, out of South Dakota, across the 4 years, I collected 80 samples. Nebraska was 73, Kansas 21, Minnesota 10. And then we had 3 samples from Missouri and 1 sample is Michigan, kind of as outliers with a longer historical record of tar spot. So basically what I saw is there is no difference from what's in the Great Plains to what's anywhere else at this point in time, as far as anything that I have collected. So that's just something that we need to be watching as we go further with studying and managing tar spot. Just, kind of be keeping an eye on that, being able to make the good management decisions that we need to, and, being able to see whether or not there are genetic differences might aid in, if there's fungicide resistance starting somewhere, the genetics might be able to tell us that, and we can better manage at that point. That is my brief little intro for South Dakota here.
Sierra, could you go back to your maps, please, real quick? People were asking what the colors represented.
Yes, so the yellow is where tar spot was found at in that year. So each individual year, the yellow is where each county was positive. The gray is where it had been found in that county at some point in the past, though that might have been as far back as 2015.
And if you're in South Dakota, you can see that 2024 where we had a massive spread of the disease across the state.
Yes.
Okay, well, that is everything that we have for an update on South Dakota. So we will go ahead and let Connie Strunk start with her talk about symptoms of tar spot. Thank you, Sierra.
All right, the next few minutes, we're just gonna kind of visit a little bit about what tar spot looks like. So for some of you, it's gonna be a little bit of a refresher. Some of you might be seeing it for the first time, all depending. So with that, symptoms, you may see some yellow flecking on the leaves or husks, but that can also be attributed to some of the other diseases, but you may see that yellow flecking, so it's just a little yellow kind of spot starting on that leaf, or you might start to see some mild chlorosis yellowing taking place on that leaf. So that's like the early symptoms. But what it's more known for, or what you typically have heard and seen, would be the tar spot or those stromas form on the leaves, husks, stalks, anywhere where there's that leaf tissue, if you will. So what those stromas look like, they tend to be black in color, maybe raised, they may be elongated, often visible on both sides of the leaf. And the big kicker here is that they cannot be rubbed off on that leaf. And we'll talk about that here in a second. As those lesions, as those tar spots, as it gets a little bit older and we have some more humidity, you may see some of the pycnidia start having some orange mass, just showing that things are kind of reproducing and continuing to spread. But we'll show a picture of that here in a second. In South Dakota, we've seen the flecking and we've seen the black tar spots. What we have not seen here in South Dakota has been the fisheye. And I know not all of the states have seen the fisheye lesion, which is a tan to brown color that will start to develop. So if you kind of imagine a fisheye, right, you got that dark center, and then there's kind of that circle around that dark center. That's the same thing that can be seen or observed in some states with tar spot. So just kind of breaking down, the flecking and the chlorosis. If you look on the leaf here, you're starting to see the yellowing occur. You'll start to see some yellow, and then you're starting to see where like the black tar spot is starting to raise up, if you will. So that would be the flecking and the chlorosis. Sometimes, it could be misdiagnosed. I mean, a lot of this can always be misdiagnosed for other things. That's where scouting and diagnosis is highly important. As we look here at the coveted tar spot symptoms, first off, we had a pretty heavy infestation of southern rust this year in South Dakota. So you have to look past that to see those dark raised spots. As you start to see, they start out pretty small. Then as time progresses, they get a little bit larger. They start to elongate, as you see at the circle at the top of the leaf. So they really start to see kind of one growing almost looks like one is growing on top of the other here at that tar spot lesion at the bottom of the leaf that I've got circled in there. But as you're out scouting and taking a look, when I talked about they can't be rubbed off, oftentimes those really small spots can be misdiagnosed or often confused with like insect frass. And so like to say, kind of wet your thumb or have something that would kind of rub on that spot. If you can rub it off, it's not tar spot. Now, if it does rub off, I would encourage you not to probably relick your thumb and whatnot, but do as you will with that. But if tar spot just cannot be rubbed off the leaf. As it gets going or ages a bit, I have a photo here underneath magnification showing the tar spot with that orange mass. So it's starting to reproduce, going to spread. Okay. Photos are all taken from and provided from the SDSU plant pathology team of staff of graduate students and workers within Dr. Shires' lab. So I just want to credit all of them. Again, this is a photo that was taken down in Missouri, not in South Dakota. So this is showing the fisheye appearance. So you see all the black stroma of tar spot throughout that entire leaf. But if you start to look at the area that has that tan kind of brown color around that black stroma, that would be what we were calling as that fisheye. You also see some of that, yellowing going on also. So again, fisheye appearance, not been found in South Dakota as of yet, but it is a symptom that some states and others are seeing. And this is just a conglomeration of photos taken here in South Dakota of what our tar spot has looked like in 2024. So we had leaves in areas that had one or two tar spot stromas found throughout the plant and throughout the field, to a little heavier infestation where it was really colored, really could look drastic. As you'll learn here in South Dakota, we did not have yield limiting with tar spot, but if entire fields started to look like this, things could have been perhaps a little bit different, but these were just a few spots within fields. So again, different pictures of what things look like here in South Dakota. As we look a little closer, again, it can be on the leaves, it can be on the husks, it can be on green and growing tissue. You can see it on the maturing or drier tissue because a lot of that chlorosis and whatnot also have colored and changed the shape of the look of the plant. So that's a few pictures here. The one here on the left is pretty striking with really, dried down, showing all of the stroma compared to a green leaf growing with the stroma on it, but also showing the tar spot lesions kind of on the bottom right of that corn leaf. Again, the one on the right was not taken in South Dakota, but just kind of showing that we did have some incidents and severity out in the field, quite similar to what they've been seeing, where they had some of that fisheye and other tar spot infestations. So with that, I'm just gonna end with my contact information, and then I'm gonna turn it back over to you, Dr. Shires.
All right, awesome. Thank you, Connie. Our next speaker is going to be Jose, talking about the tar spot disease cycle and its host range.
Thank you, Maddie. I'm gonna share my slides in a second. All right, good morning, everybody, and Thanks, Maddie, for inviting me to present today. I'm Jose Solorzano. As I said before, I'm a PhD candidate in the Department of Plant Pathology at the University of Minnesota, and I work with Dr. Dean Malvick. So today I'm going to present on different projects that we have developed to understand the tar spot disease cycle, including the host range of Phyllochromitis. So for background, our goal for understanding the tar spot disease cycle is to identify key elements of the disease that can serve as the basis for prevention and also management and control of the disease. So to understand the tar spot disease cycle, we need to look at the tar spots. And as Connie was mentioning before, these are key diagnostic characteristics of the disease. And we technically refer to them as stromata. So the tar spot, most importantly, they are a result of the successful completion of the tar spot disease cycle. And this can happen multiple times during the infection time or the disease cycle, which means that at the beginning, you will likely see smaller tar spots, as I'm showing in these pictures here. And then these smaller tar spots will become larger later during the disease cycle. And then those larger tar spots will be visible later on. And then you might be able to see smaller tar spots later on. And this is because of the polycyclic nature of this disease. So, and as Connie was mentioning here, here in Minnesota, we have observed the fisheye lesion, which is this necrotic tissue around the black spots or the tar spots. This is what we call the fisheye lesion, just for background. So in our journey to understand the tar spot disease cycle or how this happens, we initially wanted to understand what are the sources of inoculum, how does the inoculum survive, what are the environmental conditions that are required for the disease to develop, and how long it takes for us to see or for the disease to develop and spread after successful inoculation. So we started looking at the available information from previous studies, one from conducted in Mexico in late 1990s. So as Connie was mentioning, tar spot was found in the US in 2015, but tar spot is also present in other countries like in Mexico and in countries in Central America and South America as well. So we started looking at these studies from Mexico and from the late 1990s. And another study conducted in the US after the disease was found here. And we learned that Phyllochromitis, the pathogen that causes the disease, will survive in infected corn residue. And this inoculum will likely infect susceptible hosts, corn plants, under environmental conditions that are conducive. For example, more than 7 hours of lip wetness at night and relative humidity above 75%. And temperatures between 16 to 23 Celsius, roughly 60 to 73 Fahrenheit. This is what we knew about the disease. And so when all these conditions were met or are considered to be met, the disease will progress and the tar spots will show up in the corn plants. And that's how the disease cycle is completed. So while this information was great for us, and this was the available information we had before, we couldn't replicate this in our control environment studies or in field environment studies as well. So we had to fill up some gaps of knowledge that we have. And for that, we developed two different studies, namely a study to induce the disease or to complete the disease cycle in control environments, in greenhouses and growth chambers as well. And also another study to complete the disease cycle in the field. So we conducted artificial inoculations, and from these two studies, we learned different things about the survival of the pathogen, the inoculum collection, the environmental conditions, and also the duration of a disease or the disease cycle. And from that, these are some of the things that we learned. We learned that Phyllochromitis, the pathogen, can remain infective at minus 20 Celsius for up to 11 months. So we used inoculum we stored minus 20 Celsius, and we observed that the fungus or the pathogen will remain infective during this time. We also learned that tar spot can develop without prolonged high humidity and leaf wetness. And this is from observations from our field environments. We learned that the temperature Well, we have variable conditions. We had a maximum of 35 and a minimum of 5 Celsius. And just for background, we conducted this experiment in 2023 and the duration of the disease cycle was from July through September of 2023. And this is when we collected this environmental data. So we had a maximum of 35 and a minimum of 5, but the average of the temperatures at the time were similar to those that were previously considered to be conducive for the disease. Now, the relative humidity, we had also a variable relative humidity during the growing season, but the average of the relative humidity was below those 75% that were previously considered to be necessary for the disease to develop. So we have lower relative humidity throughout the whole growing season, most of the time. The dew point temperature, and this is just the temperature at which the air becomes saturated with humidity. This was similar to a previous study that was published in 2023 by Webster et al. And this was surprising for us because it's great to see that our experiments in the field were similar to those that were previously published as well. Now, one of the most remarkable observations from this study was that tar spot developed under a growing season that was mostly dry. The largest rain event we have was 45 millimeters, which is roughly 2 inches of rain. And that was the largest rain event we have during that growing season. So that's why we infer that tar spot can develop without prolonged high humidity and leaf wetness. And from these studies, we also learned that tar spot can develop, or the tar spot can appear in the plants 10 to 19 days after inoculation, and that the pathogen Phyllochromitis becomes infectious when the ascospores are produced. The ascospores are these structures that look like they're oval shaped, and these are the propagules of the pathogen. And this can happen roughly 17 to 22 days after inoculation. Now, conducting these experiments, we have learned things about the tar spot disease cycle and about how the polycyclic nature of this disease, meaning that this can repeat itself various times, may repeat itself various times during the growing season. Another thing is that there are still gaps that we're trying to fill or to obtain information for the disease cycle of tar spot. And one of those is understanding whether the pathogen can infect other plants that can serve as pathogen reservoirs during the growing season. And that's why we are investigating the host range of Phyllochromitis. Now, I started conducting tar spot research in late 2021 when I joined my lab. And during this time, we have been collecting samples of tar spot of corn across Minnesota. But we have also seen some grasses with tar spots. We have seen tar spot on grasses next to cornfields, tar spot and grasses in cornfields, and we have seen grasses with tar spots in non-agricultural settings in Minnesota as well. We have also received reports of grasses with tar spots outside of the state of Minnesota across the Midwest where tar spot of corn is found. So this raises the question, can Phyllochromitis infect grasses? Now, the truth is that across the range where Phyllochromitis or tar spot is found in the US, there are other Phyllochromis species that are known to infect grasses, and these have been present in the US for many years before tar spot of corn was found. So this raises another question, can other Phyllochra infect corn? And for these, we have been conducting a survey-based study to identify alternative hosts and other Phyllochra species that may be associated with corn. And as Sierra was mentioning before, this is the tar spot geographical range where the disease can be found. The orange represents where the disease was found in 2024, and gray represents where the disease was found in previous year, but not last year or reported last year. So across this range, we're conducting this survey-based study, collecting samples of tar spot of corn and also samples of tar spot that had been found in infecting grasses. For these, we are conducting a molecular studies, inoculation studies as well. And some of our preliminary data indicates And when I say preliminary data is that we have been conducting experiments, and this is some of the data that we have thus far. So through phylogenetic analysis, we have identified that Phylachromyces is present in corn only, and that Phylacra species found in grasses have not been detected in corn. And phylogenetic analysis is to understand the relationship of different tar spot samples that we have collected across the Midwest and in the US, across all these ranges I mentioned before. So we haven't found Phylachromyces in other plant species. We have found Phylachromyces in corn only. Now, the other step for us is to conduct inoculations. And for these, we have selected a pool of plant species, namely grasses, because these are closely, that may be closely related to corn, but also plant species that are found across this range in the US, and some of the plant species are known to be infected by other Phylacra species. We also are inoculating grasses that are found across Mexico and Central America, namely Guatemala, Honduras, and Nicaragua. And these grasses are known as Teosinte, and these are close relatives of corn. We're inoculating them because we want to know if these closely related species and other plant species that are found in the US can serve as reservoirs of Phylachromyces. So from this study, we have a pool of more than 20 plant species, and we have conducted inoculations in the field. We have conducted inoculations in greenhouses as well. Last year in 2023 as well. And this year right now we're conducting inoculations in greenhouses and growth chambers as well. And we'll be conducting research in the field as well for inoculating different populations of plants. From our results that we have thus far, we have learned that all corn relatives that are found grown naturally or wild in Mexico and Central America are hosts of Phylachromyces. When I say hosts, is that we have observed the development of tar spots in these plant species. So these are different taxa. Some are different Zea species. So corn is Zea maize, and some of these grasses are Zea perennis, Nicaraguensis, Diploperennis, different species of Zea. And some of these, like Mexicana parviglumis and Huehuetenangensis, these are subspecies of Zea maize. So we observed the development of tar spots, and this is how we have been putting a check mark on those species saying, oh, these are hosts of Phylachromyces. We have more results that we're still processing, but this is the information that we can provide to all of you right now, and be mindful that this is unpublished data as well. So by conducting inoculation developing inoculation methods to understand the disease cycle, we have learned that the fungus Phylachromyces can survive at low temperatures, minus 20 Celsius for up to 10 months. And the other thing that we hypothesize is that some of the alternative hosts can be those corn relatives. We don't have those growing naturally in the US, but those are found in Mexico and Central America as well. And we consider that those may serve as alternative hosts of Phylachromyces. And this inoculum will infect corn or a susceptible host under conducive environmental conditions, as I mentioned before. But tar spot can develop without prolonged high humidity and leaf wetness. We have also learned that tar spot can form 10 to 19 days after inoculation, and this pathogen becomes infectious or can infect new plants around 17 to 22 days after inoculation or after successful infection has occurred. With that, I would like to thank all the people that have been collaborating with me recently. These are the people that are working with me on the host range analysis, either discussing ideas, having conversations, or providing samples to us. And I would like to thank the Minnesota Invasive Terrestrial and Plants and Pest Center for the funding, and the USDA ARS National Plant Germplasm System for providing seeds for some of these analyses. Thank you.
Thank you. Great presentation.
Thanks.
All right, our next speaker, we're going to be staying over in the Minnesota area with Dean Malvick talking about tar spot situations in Minnesota.
Okay, welcome everybody. Hello. Everybody can hear me okay? So Jose just presented a nice summary of a lot of the work he's been doing here in Minnesota, answering some questions that we think are pretty important. I'm going to give a bigger, broader picture of the tar spot situation in Minnesota at a bigger scale. So, we see all ranges of the disease here in Minnesota, all the way from very low levels to very high levels, like on the right, like we do in most states where this disease occurs. I bring this slide up to make the point that a lot of what we see in Minnesota, especially in the newer areas where it's been found, has been at very low levels, like you see in that very left picture of a corn leaf. Just a small smattering of tar spot stroma on the leaf, maybe even fewer than that. And I'll emphasize that more in a moment. And, we see it mostly the normal spots like you've already heard about. But at least last year, we've seen more and more of these fisheye lesions like Connie talked about. And those pictures of those fisheye lesions are shown here on the right. And it's, it's intriguing and interesting to a lot of us because there's been a lot of work trying to figure out what causes these different types of symptoms. And no one knows for sure. There are some clues, but nailing that explanation down has been a real challenge, as others on the call can talk about, who's been more involved with some of that work. But of course, what's significant about this is that we need a lot fewer infections with the fisheye lesions than the tar spot typical lesion to get the equal amount of leaf damage. So still, the fisheye lesions is a minority of what we see in Minnesota. Most of what we see is the left, but I'd say last year we saw more and more of what we see here on the right. Now, a bit of the history. So it was first found, tar spot was first found in Minnesota in fall of 2019. And that's in southern Fillmore County. You can see in the lower part where there's that purple star. So it was first found. And what was interesting about that is certainly we've been watching for it because we'd seen it getting closer and closer from the south and the east. But that wasn't on the edge of a field. That was about 50 yards from the edge of the field. And there was nothing between the edge of the field and that first area. And you may wonder, how was that ever found? Well, the managers of that land were scouting that field for some nitrogen strip trials, and they happened to see it. So that's how it was first found. We went down and confirmed it. And then, of course, since then, it's been spreading more and more. It was found in 4 counties that year. You can see those in that kind of tan color. In the yellow color, it was found in more counties. And 2021, it spread further, although there were a lot of gaps, as you can see on the map. Now, a big question I think we have here, were they real gaps or just confirmed gaps? I think there was more out there and people didn't notice it because at the low levels, unless you're really looking closely, it's very easy to miss it. Now, moving ahead to a year ago, 2023. It was essentially a drought year across much of Minnesota in our region. Tar spot didn't develop very much at all. So this map again shows the 2 colors, the gray counties where it had been found at some time in the past, a previous year, and the yellow counties were the only counties where we found it in 2023. Now again, I suspect it was further, but it was very hard to find in the fields we looked in. In many of these fields, it only developed very late in the season, late September in many cases. So again, I think this was largely driven by the drought. And everybody would wonder, of course, well, how does this affect the potential spread for the future? Now we know already from the maps what's happened. It has spread much more, spread across all of Minnesota and more, southern Minnesota, I should say. Past the southern half, it's gone further north. As you also see, it's gone all the way halfway across South Dakota and much, much further. The good thing is, like I said, most of that area has very low levels of tar spot. That does indicate the pathogen has spread there. We know the pathogen can overwinter in Minnesota. We know the risk is probably enhanced in the future because the disease has been there and is there. But what that really portends for the future, I think is very uncertain for some of those areas that tend to be less wet than a lot of other parts of the state. The other thing I want to point out for 2024, just this last season, the only place it was really severe was that area that I've circled there in that red circle in the southeast. That's a Goodhue County area. Now it developed in other areas, including areas south of there what had been severe previously, but that was the only area that we actually had reported yield losses that were widespread. So 50 miles south, where it was severe in 2022, it wasn't nearly as severe this year, even though there was plenty of rain in both places. So it would be interesting, and I haven't taken the time to look at the weather patterns and how to tease those apart, but I think a lot of us would have predicted more further southeast, where we saw more disease in the past. Again, the risk factors, I think we're learning a lot, and we may hear more about that in a few minutes with some of the risk models and the risk assessment apps. And certainly we're learning that moderate temperatures are important, average relative humidity, within a moderate to not too high range seems important. And it's possible that too much rain may actually not be beneficial for the disease. And the other thing I want to point out is if we look at the disease cycle, it doesn't really show much about wind spread from other areas. And I suspect, based on the patterns we're seeing in the spread, that windblown spores are probably a key part of spreading this pathogen across the Midwest, at least across western Minnesota and South Dakota and North Dakota. Again, I think the disease was probably further west than we knew before at low levels, but nonetheless, it certainly spread quite a lot this year. And again, I want to make the point with this picture here on the top, especially showing how the disease develops, initially with a few large lesions as they progress. Then we get the secondary cycling of the disease and getting much more damage to the leaves. And so that reflects a lot of the challenge with both managing the disease and understanding how much damage it will cause, because there's more and more data coming in showing that if infection doesn't occur early enough, we can't get enough of that secondary infection to cause a lot of damage, perhaps. So when we see the disease develop late, like we have sometimes in Minnesota, the risk of significant damage goes down. Again, still many unknowns, although we're learning more and more. And again, I guess I've emphasized this, but an early start for the disease seems to be important. And a few summary points from Minnesota. The disease has spread now across all of southern Minnesota plus since 2019 when it was first found in the state. It's at very low levels across most of that region, fortunately. The disease risk across much of that region is still uncertain based on the drier weather patterns historically across the western part of the state. Severe tar spot and yield loss due to tar spot, fortunately, has only occurred in southeastern Minnesota, to my knowledge. Yield losses of 50 to 100 bushels are pretty well confirmed in some of those areas. And disease development has been very inconsistent from field to field and year to year, even in areas where the disease has been prevalent and serious in previous years. And that summarizes what I want to cover for the situation in Minnesota. And again, I want to thank the Minnesota Invasive Terrestrial Plants and Pest Center for funding, as well as Minnesota Corn for providing some of the funding for some of the survey work. And I will stop there.
All right, awesome. Thank you, Dean. And with that, we will move to our next presenter. So we're going to move to the southwest to Nebraska and let Tamra talk about the situation in Nebraska.
Good morning, everybody. I'll just get these slides loaded up and we'll get started. I appreciate the opportunity and thank Madalyn and her team for inviting me to share our Nebraska story. Being your southern neighbors, we are also pretty new to this disease. In Nebraska, as I mentioned earlier, we first confirmed tar spot in extreme eastern Nebraska along the Missouri River in 2021, and it was late in 2021. But we knew very early from our colleagues, some of them on this call today, like Marty, that tar spot has behaved much differently under irrigation. And in Nebraska, irrigation is really important to us. And so that was a big concern for us even before it came. And many of you have probably seen the scary picture. Thanks to Marty for providing that from Dan Heasley in the upper right-hand corner here, where you can see tar spot was much more severe under this irrigation pivot. And this is only a half circle here. So imagine it working like a windshield wiper back and forth. Well, there was disease in the corners too, where they didn't get the irrigation, but certainly you can see it was severe enough to kill plants prematurely under that pivot. But when I tell this story, and I hope Marty, you can follow up when it circles back around to you. Disease came in late into that field, and this is a good story about around this picture because it tells us about impact on yield was not nearly as severe as they expected because of how late it came in there and developed. And so we are still very concerned. And in Nebraska, we have had an sometimes impressive amount of tar spot in some of our irrigated fields, especially along the easternmost counties, where we've had the inoculum buildup over the last few years. And so this is drawing a lot of attention. And so you've seen the distribution map a few times now in this talk. And so if you focus on the one of in the lower right-hand corner and specifically on Nebraska, it's moved about as far west in Nebraska as it has in South Dakota. We have a few gaps here, but I have no doubt that tar spot is probably there. We had backed off on scouting efforts this past season as we refocused our efforts in southeast Nebraska and elsewhere looking for corn stunt. And so I think we'll continue to fill in some of those maps. And just like Dean mentioned earlier, disease severity in Nebraska has been wildly different depending on where you are. And in Nebraska, especially, it has been vastly different depending on whether it was irrigated or non-irrigated. And so here, along that western edge, as we're watching it march across the state, it's very difficult to find in some of those fields. It's very, very low severity versus some of the fields in the easternmost part of the state. It's easy to find, sometimes severe, but we're not covered with it. And so as we move forward, I'll talk a little bit more about what it's really like with boots on the ground. But first, I'll kind of show you the map because I think it explains some things. The map in the upper right-hand corner kind of shows you where we have central pivot irrigation systems in Nebraska. And we've got more irrigated acres for agricultural use than any other state in the country. And so, yeah, we really are concerned about what's going to happen here. And so you can see that a lot of that is concentrated in more of the central part of the state and in bands, depending on where we're located over the Ogallala Aquifer. We have a lot of water in Nebraska, but most of it's underground as part of that Ogallala Aquifer. And so we are watching disease in different parts of the state, but as you all know, if you irrigate especially, we have control over irrigation, and sometimes irrigation scheduling is going to impact disease development, and we're only just beginning to understand the effect of that. And it's certainly, we know that having irrigation, adding more moisture to the system increases it. But I think it's important what Dean said. I'm eager to hear what my Eastern colleagues have to say. Just like he said, having too much water, keeping leaves wet constantly can actually slow disease down. And so I'm eager to see what we learn about that. But let me show you just a little bit about what one of my graduate students has been able to unearth as far as tar spot development and impacts of overhead irrigation, that's pivot irrigation, on the microclimate inside the corn canopy. And so in 2023, it was a very different year than what we had in 24. So back in 23, it was much drier. And I'm guessing maybe some people didn't see as much tar spot in some of the other states. But in contrast, I think where we have the opportunity to irrigate, we might be over irrigating in a very dry year. Maybe sometimes we'll even see more disease in some locations because of it. And on a field to field basis, it can be very different. And so in each of these irrigation pivots, the circles, now this is not drawn completely to scale, but it gives you an idea that one of the things we're interested in looking at is disease dynamics and conditions along the length of the irrigation pivot. So just think about the irrigation pivot in the very center of what looks like the target here, stretched out across these this circle. And each one of these, the dashed circles in between, that would be the towers. That's where the tire tracks are going around. And in between the tire tracks, we call those irrigation spans. And so if I use a few words you haven't heard, I wanted you to know what I meant. At least that's what we call them here in Nebraska. And of course, the non-irrigated corners on the outside. And so my student Talon, who's now an extension professional in Nebraska too, and continuing this work as he finishes up his master's, he focused his efforts on disease ratings and also microclimate measurements with weather stations at the innermost span or the second span, and then the outer span of the pivot furthest away from the pivot point, as well as in the non-irrigated corner. And so in that first year in 23, as we were just really getting a handle on what we were doing, he had 4 locations, 3 in extreme eastern Nebraska from north to south, and then 1 just barely into South Dakota in Clay County in that southeast corner. As you can imagine, Talon has collected mountains of data over the last 2 years, and so he summarized this in a way that I'm just going to show you some snapshots of some interesting observations that he's made. And so in 2023, you've got graphs here for what air temperature and relative humidity looked like in the inner span of the irrigation pivot. That's the blue line that you see in this graph versus the outer span and in the non-irrigated corner. That's green and yellow up there. And so in the vertical lines that he's got on this map, that's showing you each line is about 15 minutes. So you can see for an irrigation event, which is what he's showing in this image here, that in the inner span, we're irrigating over a much longer period of time, over a few hours, versus in the outer span where it's moving much quicker to keep up as it moves around. And so that may only be for about 1 hour. And the biggest change that we saw was, and you expect it too, right? You would expect to see a temperature reduction. And at its maximum, Talon saw a temperature reduction of up to 13 degrees. Now, the temperature was less over as much as 7 hours during that time, but it was 13 degrees Fahrenheit less for a 2-hour period. And that's, to me, that's a much bigger difference than I expected. And that was in the inner span where we saw that biggest temperature difference. You see where that dip is in that line. Well, as you might also expect, relative humidity is also impacted as well. And so in the graph on the right-hand side, on the blue bar, you can see relative humidity increased much higher in that inner span in that blue line compared to the outer span and of course, the non-irrigated corner. And so relative humidity here for as much as 5 hours was increased up to 41%. And at its highest, at its peak, was 3 hours. And so we're seeing a dramatic change. And I think my takeaway from this is that the changes are there that I expected, but they are at a much longer duration and much greater magnitude than what I was expecting. This particular location was in Burt County. And one thing Talon wants to point out too is that we saw these biggest differences during irrigation events during the daytime. And so, we're a Great Plains state too. We're often arid, dry, windy, sometimes temperatures very hot too, sometimes in the 90s. often hitting 100 degrees, which we did in 2023, that's when we saw those biggest differences. The irrigation events that are occurring in the evening hours, when everything is more stabilized or normalized across the pivot, the changes weren't as different. Moving on too, he also measured vapor pressure deficit. And so that's the difference in the amount of moisture that's in the air versus the amount that it could hold at saturation. And our irrigation engineers that we've really enjoyed working with, they really like this measurement, and we're actually surprised that the difference in the vapor pressure deficit was as big as it was. This is a common measurement that they use in irrigation research. And here you can tell the same thing happened, a huge drop in vapor pressure deficit, different in that inner span versus in the outer span. And then, we start thinking about other things, other conditions in the canopy, and you've already heard about the fungus and the type of conditions that favor the Phylochoromatus fungus and infection and disease development. Well, we know leaf wetness has an impact as well, and there's still more for us to understand. But just as you might expect, in that inner irrigation span, the leaves are wetter for hours longer than they are in that outer span. And so as Talon began to evaluate disease incidence and disease severity out there in the field, the example he shared with me was in Saunders County in east central Nebraska. Early on, he was able to find tar spot in this particular field on August 9th, for instance. And the incidence, that's the number or percent of plants that have disease visible, was 40 to 60% more under the inner span and even some in the outer span too than in the non-irrigated corners. And when he monitored disease severity over time, it did increase faster than what we saw in the outer span and rain-fed area of the field. But in this case, it was not always statistically significant. But looking at the bar, you can see sometimes we did see some impressive and more severe disease development over that about a month after that. His research objectives have changed a little bit as we've tried to redesign how we are collecting these data. And he's doing a lot of field work, and some of these conditions just have not made it easy to gather the type of data that we need. And so this is a picture of his weather station there in the canopy, and he's begun to measure conditions at different levels in the canopy, and also trying to get data from more fields across the eastern part of the state where we've got more disease. And so when we talk about different levels in the canopy, we're talking about focusing on the ear leaf and then 2 leaves above that and 2 leaves below that for disease and for conditions. And again, measuring some of the same things that we measured in that earlier study. One thing that was interesting for us, and he's highlighted this from his location in Colfax County, was that disease was different. And you saw that in the earlier work in 23, where we had often more disease closer to the pivot point. Well, they saw that again in this other study. And I should point out too, the design was different, where instead of walking into just those 3 points into the pivot, he actually had a number of fields where he was making line transects across there at random locations, and then collecting data on disease development at, I think, 60 total points in the field. And so you can kind of see where those points were, and using GPS, we calculated the distance he was from the pivot point in those fields. And so then he was able to generate these graphs. And kind of like what we saw in the earlier work, disease was more severe closer to the pivot point and diminished as he got further from it. But it also mattered where on the plant he was evaluating disease severity. On the lowest leaves, that difference was much bigger. And you can see that in the orange graph on the bottom here. And so disease was much more severe in the lower leaves, closer to the pivot point, and it dropped off. And as you moved up the plant, those changes, weren't as dramatic, but they were still there. And so that was something that's changed our scouting recommendations here in Nebraska. We recommend people look around the pivot point in those first couple of irrigation spans when looking for early season disease to see if it's developed. But that has not always been the case, and we saw differences between 2023 and 2024 that I think probably had more to do with what our actual weather conditions were. And so if you think back about last year's conditions in 24, out here in the Great Plains at least, we had a lot of rain in Nebraska in that first half of the season. And when we got to July, it's like the water was shut off. We stopped having rain, and thus we started irrigating a lot. And it probably impacted what we were seeing. And so when he visited other fields, and he's visited 25 different fields, and you can see in the map on the bottom where those pins are placed, where he was scouting. And most of those in the easternmost counties of the state, because he needed to see disease and estimate that to get a better idea between the inner span, outer span, and again the rain-fed corners. So reverting back to this other design I shared with you. And in 24, it didn't matter which span he was in. Across those 25 fields where disease severity varied greatly, from barely being able to find it to being very severe in a low percent of those fields. It just mattered that it was getting watered by the irrigation pivot. So whether you were in the inner span or the outer span didn't matter. And it was all more severe than what he saw in the rain-fed corner. Likewise, though, I want to bring up, I think it was Connie that reminded us, tar spot probably wasn't our most important disease this past season. It was the flush of southern rust that made it up here into eastern Nebraska, and especially in northeast Nebraska, closer to South Dakota. We had some of the worst southern rust we've seen since 2006. It created a lot of alarm. A lot of fungicide applications were triggered. But a lot of the concern that happened here was that a lot of people might not have been aware that rust fungi, southern rust and common rust, switch over to, instead of producing the orange and familiar red spores that we're used to looking at, they transition to Telia spores toward the end of the season, and those are black spores. And so we started seeing black spots on those same leaves where we saw so much rust. And this was in areas where we were seeing tar spot too. And I think people's perception of what was happening with tar spot was different, because there was a lot of misdiagnoses on the ground here that they were thinking that it was tar spot, and it turned out to be that a lot of that was southern rust instead. And so I wasn't watching the time very closely, and I haven't watched chat at all. I see we've got, looks like there are a few in there. I don't know if any of that's been tackled by any of my colleagues or not, but I'm always here to help if I can do anything. I live in northern Nebraska and travel the state quite a bit, and so I care what's going on in South Dakota too, and keep up with folks over there. So thank you all so much for attending today, and good luck this season.
Awesome. Thank you, Tamra. All right, so we've heard kind of the situation in South Dakota, Minnesota, and now Nebraska. So we're going to kind of move into the main part of our program, where we're going to have Darcy, Damon, and Marty talking about their experiences with tar spot, kind of, in those areas that have been dealing with it a lot longer than we have. And that way we have the opportunity to kind of learn what they've gone through and their lessons. So with that, I will let all three of you take it away.
Thanks, Maddie. I didn't get a chance to actually introduce myself early on, so just a short introduction to myself. I'm Damon Smith. I'm the Extension Field Crops Pathologist at University of Wisconsin-Madison. Like Marty, we've been dealing with tar spots since 2016, so almost a decade now of experience. We've had several really substantial epidemics here. And probably unique to some of us, Marty and I, and maybe Dean as well, we deal with not only grain corn, but also silage corn. So I've got experience in both of those systems. This is going to be a little different than the previous slide deck, so I'm going to drive the slide deck. And we're going to be more conversational here because there's three of us that are going to be presenting. So we have a shared deck here, which I'll get pulled up here, and my colleagues can let me know if it shows up okay. Look all right there.
Yeah, you're good.
Okay. So we're just going to spend some time today to dig in on kind of what we know about management practices and some other things, and we'll be kicking stuff back and forth. So Darcy's going to actually get us going here and just tell us a little bit about what she's seen and observed in Indiana.
Okay, well, thank you very much, Damon. Good to be here. So what I start with, and this is what I've been talking with all winter, is this opening slide here on tar spot, where I'm looking at some severe infections. And what I like to share with our growers, and so in Indiana, we've had some severe epidemics, and I'm coming out of 2024 where we probably lost about 5% or more yield to tar spot across the state. I had areas that experienced that 100 bushel acre losses where they just didn't get management tools out there. And then in some areas, the disease did not act like it did every other season. So my first bullet point when I talk to my guys here in Indiana is every year has been different since we've been fighting this beast since 2018. But as we talk about it, what I like to talk about is you see this leaf on the left that's green that has about 5% or more tar spot on that. If that's at the ear leaf and above, I'm going to tell you it's game over to try to implement any management tactics. We can find the disease earlier to make a decision, but once you've reached this point, we're really not going to slow the disease down. And in Indiana, when we have favorable conditions, we can go to the disease on the right, that blighted leaf, within 2 weeks. And so the yield impact's all going to depend on when this is happening, in what growth stage that it's hitting. So if the disease does start earlier, then we're going to have some significant losses. If it doesn't move into and we're at dent when this all starts to happen, then we're going to escape some of those losses and really not need to implement a tactic. So that's some of the information that I like to share year to year that it changes. And I think, David, you can move on to the next slide.
Yeah, so I think there was some discussion earlier on just about like the incubation period and stuff. So if you could take a moment to kind of talk about that and how it relates to some of the biology.
Right. So I like to show our growers what's going on a little bit in these stroma. And I know they talked earlier about some of the infection. So what I'm showing here is just, what the stromata look like on the leaf surface. If we zoom in, a spore-bearing structure there that's releasing those ascospores that are the primary infection points there for tar spot. And so what we've done is a colleague down the hall here has done some microscopy work on looking at this infection. And I think that's the next slide, Damon, if you have that. To show how the infection looks like along the leaf. And so if we look at A, that is a healthy leaf, no infection point, you have an open stomate there. It's a clean, healthy area of the leaf. But as we move in, B is showing where we have that spores germinated and we have some mycelia infection occurring around that area. And then it's going to continue to develop. So about the C picture, we're starting to develop that black stromata, the black spot that we see. And it does take a long time. And I still think we're seeing 14 to 21 days from that spore landing to us seeing some of those first stromata develop. And then as it continues to develop, what we're showing in the rest of the pictures is the different spore-bearing structures. So there's both asexual and sexual spore-bearing structures. I know as farmers, you really don't need to worry about it. But what I really want to show you is you see this is just looking at one black spot. And you can see all these structures that are forming there, and they're consuming all that leaf tissue. And then as they develop, then the spores mature, they're going to release those spores. And sometimes we see it come out in a gelatinous mass, which will be distributed probably through water splashing, but then they're also probably releasing spores that move it up through the canopy. So I think we see infections moving down the leaf. So we have secondary cycles moving down the leaf, and then we'll have secondary cycles moving the disease up in the leaf. But I just want to show, once we have those black spots and they're fully developed, there's nothing left within that leaf material to contribute to producing that ear of corn that we're trying to form. So again, that tissue is pretty much gone once we have infection.
Nice. Thanks for that, Darcy. And I can just talk a little bit about some of the residue work we've been doing. We get a lot of questions on this. Probably, in South Dakota, it's similar to here. We have a lot of corn on corn, no-till acres. And so we looked at how does residue play a role here. You've heard all today about how the fungus can actually overwinter. in that residue, and that certainly is the case here in Wisconsin. We've looked at those systems, so this is just an example of one location that we had here in Arlington, Wisconsin. Looking at corn on corn, high residue, and we looked at different levels in the canopy where we rated for disease, and we had a couple of different hybrids, one more resistant and one susceptible, and just kind of following how the disease moves up. So as you might expect in a high residue situation where the fungus is overwintering in that residue, we do indeed get a bottom-up movement, as my colleagues have shown in some of their other data. And of course, resistance plays a role here. By the time we get to R5, we see lower levels, but it is partial resistance, so it's not perfect. Basically, what's happening with that resistance is it's slowing down the secondary increase in that fungus. So not a lot of surprises, as you might expect in a corn-on-corn no-till situation. What was a little more interesting, and we've got more data points along with this, is that we also had some cases where we had no residue. And in fact, in this field, we were actually following soybeans. So it was really hard to actually find any residual corn residue in this particular situation. And again, you see a similar bottom-up trend in terms of how the disease actually spreads, which is a little bit, it makes you scratch your head just a little bit, because what that tells us is that the spores are still continuing to come from some infield source. And so what is likely happening is you still have probably small pieces of residual leaf material. Those stroma are a very strong type of survival structure, and probably still in that soil matrix. What you can see, though, if we compare that to what you saw in the no-till situation, it does slow down the disease a bit. But by the time we get to R5, you still got plenty of tar spot, if the weather conditions are good, up in the upper part of the canopy. So the moral of the story here is that, yes, residue management could be part of it, but I would not recommend abandoning your no-till to try to manage this. I think spending time with the hybrids, and I think we'll talk a little more about hybrids here in a little bit. I think spending your time putting energy into that hybrid choice is going to be give you more bang for your buck than trying to manage the residue situation. So with that, I'm going to let Marty kind of jump in here and let him talk a little about what he's learned in some of the agronomic work with nitrogen and also seeding rates. So Marty, take it away.
Yeah, thanks, Damon. Yeah, so we were obviously getting questions as well about nitrogen management, planting populations, planting dates, all these various things. So we implemented a couple of trials over a couple of years to have a look at this. One of those is nitrogen application rates, running from very low nitrogen, 80 pounds to the acre, up to 240 pounds. And then a planting rate as well. So low plant population, and then up above economically optimal planting populations. And we use a couple of different hybrids as well to incorporate that element of difference in variety, susceptibility, or resistance. Next slide, Damon. So here's the data slide for the nitrogen work. So in short, there was no difference by the nitrogen rate. So either looking at 80 pounds of nitrogen applied as urea as a side dress, all the way up to 240 pounds. We weren't able to push disease with high nitrogen like we may expect. We certainly do see that with some diseases under high fertility. We tend to get more disease take off. But with tar spot, we weren't able to drive that. There certainly may be more complexity to this, and maybe under some situations, yeah, you can drive things a little bit, but it's a pretty minor effect. What I do want to point out is the effective hybrid. So at each of those locations and every single year, hybrid is what drove the response. And I don't think we can emphasize that enough. And as tar spot moves into South Dakota, that's going to be your challenge, identifying the hybrids that are really susceptible and trying to move them out of the lineup and pulling in those varieties that will have more resistance to disease. So next slide, Damon. So the other thing we looked at was plant population. We deal a lot with white mold. Damon does as well in soybean. And so typically we get more disease under high plant population. So, talking with my students and whatnot, like, what do you expect is going to happen when we do this? What do you think is going to happen? So we'd probably suspect that this higher plant population would drive moisture in the canopy, microclimate, perhaps, exacerbate disease. Next slide. But we found the opposite. So this was really interesting, a good sort of teaching moment too. So I've got here the resistant variety on the left-hand side, susceptible on the right-hand side. So again, variety is key, big differences in terms of susceptibility and the amount of disease. But you can see very clearly, especially in that resistant variety, the low plant populations, we actually recorded significantly more tar spot than that high plant population, which is really intriguing. I think it does come back to microclimates still within the canopy. We have some more data that we're working through at the moment, and what we suspect is happening is that at that low plant population, we're losing radiative heat from that canopy quickly and then setting up leaf wetness on those plants and potentially promoting infection. That's one hypothesis. The other is, potential of more air movement, turbidity within the canopy at lower plant population. So it's probably going to be a little bit hard for us to pull those two things apart. And that's where, the work that Jose was talking about is really helpful to try and, really get at exactly what's driving disease. And all that comes back to improved disease modeling too. But anyway, really interesting. Bottom line here, go to the next slide. Make sure you plant at your economically optimal seeding rate. So you could, this is good news for seed dealers, right? Like, yes, plant more seed, you'll get less tar spot. Well, that's not economically viable either. So out of that data, we looked at the economically optimal, and it fell exactly where it does, regardless of disease. So keep doing what you should be doing for best agronomic and economic performance.
Yeah, it's interesting you bring the population thing up, Marty. I've done some work in silage as well, and we saw lower planting rates, which corroborate your evidence with higher tar spot. And then the other place that I see this, we have a lot of sweet corn here in Wisconsin with our processing crops, and those are notoriously lower populations than you would see, and boy, it just hammers low population sweet corn stands when the environment's really good. So I also recommend, at least on the dry land acres, which is a little different than what Tamra talked about, but scouting, I go to the edges first actually to look for tar spot, which is sort of counterintuitive. I don't know, Darcy and Marty, where do you guys tend to go when you should roll into a field?
I'm still going the edge, but that's also easiest. But I may look for under a tree canopy where it's a little cooler, but along the edges, and then that's kind of where we start, and then decide how far we can go in if we're not finding it before we quit.
Yeah, I agree. And sometimes the western edge, because that doesn't get that morning sun. So I mean, it's just anything that might be collecting air, prevailing winds, I guess, and anything that's going to be driving any leaf moisture, like tree lines at Darcy or low areas and gaps.
So I guess, Darcy, could you expand a little bit kind of on what you've learned with hybrid reaction and what we know about susceptibility and resistance?
Yep, Damon. So all winter, I just went back as we're getting all these fungicide questions, and we'll get to that. But I want to back up and just say, first and foremost, if we find host resistance, we backstop that and we won't rely so heavily on our fungicides. And I just wanted to, from day one, we've seen a different reaction across the hybrids across Indiana and other regions. And this is kind of what we see in our plot. So what I'm showing is two hybrids that we have selected that we do some of our research on, where I have my highly susceptible one, really good for every other disease. So we're really looking at tar spot. And if you can see there, what I would say is that we got really healthy stromata forming there. There's no, they're growing healthily and there's lots being formed, versus where we've selected some of these moderately resistant hybrids where you can kind of see they're there. And what I see is that that's the initial spore shower almost of what happens. So you can see how much inoculum is already landing on those leaves, but you can see that they're not growing very well. They would have what I may call a hypersensitive response. We haven't really identified what's going on there for that reaction. But I talk about, in our, we think about being infected by a disease. We have a host response, immune response to the infection in our bodies. These plants also look like they're having an immune response, keeping the infection at bay. That being said, later on the season, as the plants begin to shut down and reach maturity, I also see where these spots begin to grow again. And so the reaction's no longer there, not containing some of that infection. But again, this is what we need to look at and monitor in our fields. And so when you're trying to identify hybrids, I tell you to go out and look, cross the road and check your neighbors and see if there's a different reaction. I was in fields this year where they had sprayed a fungicide and it was still just lit up with tar spot in a susceptible hybrid. We crossed the road and they had a different hybrid. They hadn't sprayed and the disease was being kept down because it had this good kind of moderate resistance available. So that's just something you can keep an eye on. Damon, if you move to the next slide, we've done some work looking at that, trying to see how they integrate together with spraying and the hybrid susceptibility. And so what we're showing here is 3 years of data where we've combined, where we've had good disease pressure up in northern Indiana. And so you can kind of see on the black bars are non-treated. And so you can see if we added some of this moderate resistance into the system, we automatically decrease the amount of severity of tar spot in that crop canopy, keeping it below, I would say, our 5% number, keeping it at a lower level. And then when we add the fungicide on top, we definitely get the step down of protection with a fungicide in that susceptible hybrid. But if you look at our tolerant or resistant hybrids, you can see that step down is not as great. And because of that, that's probably going to change our return on investment for using the fungicide in the field. And so if you go to the next slide, Damon, we kind of separate it out by looking at the hybrid response to yield. So you can see, yes, my susceptible was my lowest, but there's still a mixed bag on the yield potential of some of our resistant lines out there. One significantly increased yield by just using resistance versus the other was in the middle of the road. So again, I think we're getting there. And as more hybrids have better evaluations on tar spot, I hopefully will start to identify some that'll be really good for this disease in the field. But overall in Indiana, what we've really found is when we have severe tar spot, yes, a fungicide, even going at the standard VTR1, does provide that yield protection that we're looking for. I know every year is going to dictate how much of that return on investment is going to be there, but we have shown, and we'll get into more of this, that yes, a fungicide when we have severe tar spot will protect some of that yield.
So just a question for you, Marty and Darcy. I hear a lot about, company X, their genetics stink. Company Y, their genetics are good. And I guess my observation, I'd love to hear what you all say. I don't think it runs that line, right? Like, I think, each company has individual genetics, which some are susceptible and some are resistant. And our agronomists like to talk about each hybrid standing on its own. And I guess I just like to hear your opinions as well, whether you see specific hybrids within companies having better resistance. And the reason why I bring this up is because I think it really is important that folks take that individual seed catalog and really study every individual hybrid and then look at what's going on with the university trials as well and really understanding that. So I'd love to get your opinions on that.
Yeah, so I guess we've seen some of the large seed companies put trials out side by side. And even though there's apparently perhaps some differences, at the end of the day, the yields were very comparable. So, and then just speaking about company X that, maybe does have better tar spot. We had an instance where company X was lit up with northern corn leaf blight this year. So it's, I think you've got to be careful, drinking too much of that Kool-Aid. I think it's really important to assess hybrids.
Darcy, any thoughts or?
And I've seen a mixed bag. So I've seen both sides, multiple companies have different hybrids. And so they need to, and that's why I tell you guys to get out and look at the hybrids you like and see if you can see differences on your farm. Don't just put everything into one hybrid, but pick some hybrids so you have some backup to make that assessment on your farm of what works good in your region. And I know some of the values that coming out of the companies has been hard to get because they see different reactions. So one hybrid looks really good for Indiana on tar spot, and then they take it to Illinois and it flips. And so there's some of that going on, and that comes back to we're still trying to tease apart what's going on, and we may have different populations that react differently to each of the hybrids depending on what genes they've identified to increase that resistance to the disease. So I think it's still a mixed bag, but again, if you can identify some, I think that's a starting point. And, I think about some of the diseases out there that we manage really well without using fungicides, and I hope 10 years from now, we're not doing this anymore. We're not doing this part of our job anymore because we've discovered some good resistance for tar spot. So that's where I hope we'll be. And I would just, look at and assess and talk and watch the season and know that it may change. It may look good early on, and then as the plant shuts down, everything may move in and they all look the same at the end. So again, it's going to depend where you're watching it.
Yeah, resist tar spot tunnel vision is what I tell farmers in Wisconsin, right? Like, I mean, we had like you, Marty, we had like epic northern corn leaf blight last year, and everybody was hyper focused on tar spots. So, you know, just keep those other diseases really in mind, you know.
Yeah. Can I make a brief comment from Minnesota?
Yeah,
sure.
You know, we certainly see differences in hybrids for tolerance or partial resistance to tar spot too. But consistency and overall reliability, I think, is a good thing to question, because certainly most seed companies have had a hard time to get repeatable data from multiple seasons because the disease tends to be inconsistent. So somebody's looking to buy a hybrid specifically for tar spot, and that's the primary reason for their choice. Make sure to ask questions like how much data is behind that choice.
Yeah, good point, Dean. Thanks for pointing that out for us. So you've already heard a lot about kind of the environment and what's going on, and it's great to see a lot of these follow-up studies to some of the work that had been done across the U.S. Just looking at following tar spot. This was actually some work that came out of the National Predictive Modeling Tool Initiative, which is a federal initiative that comes to the Ag Appropriations Bill every year, and we had set some trials out over the past years, hitting some good epidemic years and some not so good epidemic years, but we were able to actually look at a lot of small plot trials as well as commercial field trials to gain some data just on how does the weather affect tar spot. And you saw from Tamra's work and Jose's work, you know, we're starting to really understand that these fluctuating conditions and especially temperature seems to be a really significant driver here, along with some level of leaf wetness. But it's not rainfall, like extended continuous rainfall. That actually seems to be a bit counter to that. And we saw that a bit in some of the modeling. So we took all that work that we had done over those commercial fields and small plot fields, and I won't bore you with all the statistics here, but where we arrived at was a set of risk models, and these are depicted in the surfaces here. And so this is a probabilistic model. You think about this sort of like rainfall, or rainfall prediction. You know, today might be a 60% chance of rain. You might grab your umbrella if that's the case. And so what we're trying to do here is we actually run two models. We call that an ensemble. We then find the average agreement in those models, and then we piece that together to give you a risk probability. So today might be a 60% chance of tar spot. That means you probably need to start thinking about some scouting, looking at fields with susceptible hybrids, maybe thinking about a fungicide application if you're in the right growth stages and those sorts of things. But what I want to point out, and others have pointed this out with their work that was presented earlier today, it's really temperature and leaf wetness, not continuous rain events. And we depict that in both of these models, and really temperature is the driver. And it was really interesting to see Tamra's data with pivots and looking at the variation of temperature actually across the pivot. That really kind of corroborates what we see. Temperature is a really huge driver here. And especially when you have conducive temperature in the evening, which coincides with leaf wetness. And we captured that actually in this B model here. So we use a relative humidity surrogate. So we use 90% relative humidity above when we count the hours. And so when that happens at night with those cooler temperatures, we believe that those are really good conditions. And Tamra also talked about, and Jose talked about, the extended periods of leaf wetness. So when you go out in the field at like noon in the summertime and those leaves are still wet and your boots are still getting wet, those are really good conditions, especially when you have sort of below average temperature conditions out there. So you have to keep all this in mind when you're thinking about what is really tar spot weather. It's a little bit counterintuitive compared to some of the other diseases out there. We've taken all this and we built sort of an easy tool to use. This was our first kind of go at it. It was called Tar Spotter. That QR code will actually take you to the landing page where you can find the iPhone and Android versions of the app here. It was built on an app that we had developed called Sporecaster in soybeans. Again, it's using some of these models that we had developed. We take all the sort of the math out of it for you. The phone does all the work. It hits your GPS coordinate, pulls down 2.5 kilometer grid of data to your phone, and then uses that weather data at that GPS coordinate to give you a risk probability. So it is a tool to help you kind of look at what's going on there. We're also going to be deploying this tool in a sort of more global type tool in the crop protection network this year. So look for that. We're hoping to also run some other corn tools there so you can sort of balance what's going on out there. We've done some work actually looking at how does tar spotter sort of do out in the real world. This is just an example of how we actually time some fungicide applications using that tool. So they're non-treated there on the left. We're strolling down through the field here on the right, looking at where we time those applications using a fungicide. Not clean. It's not perfect on the right-hand side. You can see plenty of tar spot in there. But we kept those plants green a little longer so that we could continue to fill out those kernels on the ear, which helped improve yield. Darcy's also looked at this. This is some great data, and she can jump in here if I mess this up. But I really like this data set here because she took a look at it on both resistant and susceptible hybrids again, and showed that yes, you know there's kind of a sweet spot really in terms of timing, and it starts about tassel, goes all the way to kind of early R4, although I think that's kind of playing with fire a little bit. I'd rather see those applications kind of go on at by R3. But tar spotter can really fine tune those applications. And where I see folks actually using tar spotter here in Wisconsin, and I'll let my colleagues kind of chime in here in a second on how they see it used in their states. Where I see it used is a lot of times our farmers are going to make that tassel application anyway to try to protect the plants. And they're using tar spotter to make the decision on whether they actually need to come back and make the second application by, say, that R3 application. So trying to figure out whether they really need that 2-pass program. So it can help sort of not only improve the accuracy of the applications, but make a hard decision there on whether you got to be more complicated really in your fungicide application strategy. I don't know, Darcy, you want to make any comments here on this data?
Yeah, so for this data, the tar spotter app triggered at R2. And so we sprayed at R2, and then it had a follow-up at R4. Mm. And so that's where that one went. So you can kind of see it pushed it. We came in late 23 was a later year when the disease started. So that was probably right where the R2 was a better timing versus the VTR1. And that's kind of where that triggered. And I think that year, I don't know if you got the yield data, that the tar spotter was the one that only pulled the yield, right?
Yeah, I didn't get.
No, you didn't put the yield data. So the yield data that year, the tar spotter, the 2-pass program was the one that netted us the best yield in the susceptible hybrid, whereas we were flat on yields for the resistant. So that even though you can kind of see that by keeping it at that 5% or less, we're really going to not see a lot of yield impacts in that resistant hybrid. But again, it's worked for us. In Indiana, we still have some issues with the second trigger coming in right, but it does help us on that first trigger where tar spotter has been really good at picking up when we should go and put that first application out. I also would use it to go out and look for it. So if we've reached, I think we turn it on at V8, right, Damon? Yeah, so if we cheat it and turn it on at V6, it's going to be probably red for all of us. But if we wait till V8 and then turn it on, it's been pretty good at saying, yes, the conditions are good to when do we need to spray for it. So you can see we turned it on, it didn't come into R2 versus those earlier applications. But some, if we're before those vegetative growth stages, it may be positive just because our conditions are more conducive in the springtime for the disease that's going to trigger the model.
Yeah, and I've seen it kind of move around year in and year out, right? So like it tends to kind of stay in that tassel to R3, but it can move back and forth just depending on when those conducive conditions line up. So it's not exactly the same every year, but that does seem to be the window. All right, so I'll hand this back over to Darcy. She's got some multi-state tar spot trials that she wants to talk about.
Yep. And so what we're going to show here is just 1 year. And what we're showing is that severe year where we had severe tar spot in all 5 states. So you can kind of see in the end, the severity at R5 is 25% or more. I'm just going to say when we reach that leaf is going to be dying. It's not going to last very long at R5. But when we combine the data, what was really interesting, I think what we see is many of the products that we have labeled for tar spot will control the disease, but there is a difference in the longevity or how long they survive in the canopy. And I think this is some of the separation we're starting to see there, is that some of these products, the Delaro, Complete, the Veltima, Revitec, persist a little bit longer, and that's why they get a C versus some of the ones up front that got a B in that. But you just note that we do have a large choice of products. And so I'd say you got different choices to pick from on your farm if you want to go with a single application for tar spot. And then looking at the yield potential of these when we have severe disease, and I'll have you click it again, Damon, I think it's animated, that we saw on average the fungicide when we sprayed it provided 3% or more of yield protection. That got us about 17 to 18 bushels per acre. If we were to run the numbers now at $4 corn at $30 an application, we need 7.5 to break even. If that was the number I've running all winter. So that's something to think about. Back in 2021, prices were a little higher, so we could tolerate a little less yield on the return on investment. But then we did have those 3 products that provided 15 to 18 bushels. So that definitely would pay for using a fungicide to protect for tar spot. And so I will comment, you can keep this going, Damon, it's fine, that for these efficacy data trials, we run them every year, and that is what feeds that efficacy chart that we have from the Crop Protection Network. So that is a really good resource. Not only does it have tar spot, it has southern corn rust and all the other diseases. So you can cross-reference and make an identification of, yeah, I want to, I'm looking at this product, how well does it do for those diseases of concern on our farm? Just not only thinking tar spot, but all the other diseases we may need to worry about. The other thing we've been looking at in Indiana is timing. Oh, back up one slide. Yep.
Sorry.
We have it animated. We've been doing these timing trials in Indiana, and this is looking at those years where we had severe tar spot. And what I've noted here is we started with these late vegetative sprays, and then we add, our standard VTR1, R2, and then we had a 2-pass program going on, a late vegetative followed by VT. As we built the Tar Spotter app, we put it into the system. It finally turned on for us in 2022. Probably that year, I will acknowledge, so the 2nd of August, that went at V12. If we go at V12, and I don't know if we'll have that data, if we go that early, that doesn't get us to dent and black layer. We probably need a 2nd pass if we're going to start that early in a 2-pass program. But it's also important, you can see that the disease started in different time frames in each field. So middle of July in 2019, end of July in 2020, and then the 3rd of July in 2021. And so each of those time frames is going to influence the best timing, their optimum timing for tar spot. And I think if you go to the next slide, when we think about spraying, I just want to emphasize what happens if we go. So we hear a lot of talk about spraying at V6. So if we spray at V6, we cover the whole plant. And I think if you click it, Damon, one more time. If we look at by V12, one more click, that's what's covered by V12. So if then we were to spray at V12, kind of where the plant gets covered, but by R1, when we're spraying the crop canopy, click it one more time, you're going to see we're only getting the top 2/3 of the canopy. But if we're talking about what's feeding that ear, these are the leaves we need to really protect when we're talking about foliar diseases in corn, or what I think about. And so I think I'll have some slides at the end to show that this disease is at least going to tell you how good your coverage is. So if we move on to looking at timing, I'm just pulling an example here in Indiana where we had the disease move in. So here what I'm showing is where the disease was first found, that straight red bar, the development of the disease over time. And I do note that about R3, things turn on for us. And about R3 is where we see that exponential increase in disease in the canopy. But if we sprayed at V8, V10, see what it does to our disease curve, it doesn't really shift it. So those 2 time frames in 2020 went out too early. By the time the disease was moving up into the upper canopy, those products are gone from activity. If we sprayed at VTR2, what I'm showing here is, so at VT, I didn't have any disease at the ear leaf. But at R2, we had about 0.3%. And what I'm showing here is an image of what 1% looks like at the ear leaf. So we had some disease at the ear leaf. If I'm finding 1% of disease at the ear leaf, that disease is throughout the field, and it's probably on every plant in those lower leaves, particularly with the fields that have had a history of tar spot. But you can see with those 2 timings, we've shifted the disease curve off. We've pushed it down the road, trying to protect that yield potential. Whereas now, if we go and look at spray at R3, we had 2% of the ear disease in that upper canopy. So you can kind of see, you watch that blue line, I'm starting to lose control. It's creeping back up towards my non-treated. We did get some yield, protection of that upper canopy that wasn't infected, but again, we're starting to lose control because there's infections we don't see. When we spray this trial at R4, there was 6% in the canopy, and I'm going to say this is a failure for me. I really don't shift the disease when we're spraying. We might see a little reduction in that at the end. There's still some green area of green leaves that aren't infected by R6, but again, this is where, and when I talk about, I will not spray, I will not recommend spraying if you missed it and you have 5% or more in that canopy. I would say game over, let's let it go for the rest of the season. And then we continued, we sprayed at R5 in this trial, which had 16% of disease. So I'm going to say that was complete failure. Now we're putting a fungicide out there, not getting disease control, and probably not protecting yield potential. So kind of that's where some of the recommendations that I look at on the optimum window. And so that, I think if you move to the next slide, we'll look at the optimum window. And so what we did is we merged the 3 years of data, because I can talk about maybe in 2019, our optimum was VTR1. And then in 2021, it was V8 followed by a second application. But if I know that doesn't help you every year, it's the crystal ball. We can't answer when do we need to spray, right? Because we don't know when the disease is starting. And so when we merged all this data from our plots in northern Indiana, what I find is, and then if you click it one more time, I show the average level of disease that was in those plots for those 3 years. And then one more time, I have this window where I get good disease control, where I want to land. So I want a fungicide not only to provide me good disease control, but then yield protection. But you can see there, so for an Indiana, VT to R3, that's the window I want to land if I'm going to put a fungicide out to control tar spot if it's active in the canopy. That being said, if we add then, what is the yield potential? What is our return on investment? If you click one more time, Damon, this will show the net return. So we've subtracted our untreated yield from that treated plot, and then also ran the numbers on the net return of spraying, and that window actually opened. And so if you click one more time, it opens up where I had a positive net return all the way to dough. And so when we get to the end, when we show our recommendations, this is where I would tell my guys, I might be willing to spray at dough, because we may still have some yield potential to protect. It may not control disease. And if I talk back, I want disease and yield protection. So I really wanted to go a little earlier, but there's some yield to protect at dough. But if you look at that Dent R5 spray, we don't get the return on, we don't get the yield protection we needed. It was way too late. And so this is kind of where our recommendations are landing on. I would generally, if I'm going to go for a one-time spray, I'm probably going to start at VT to R3 with my optimum window of that to and then expand it to dough if it's coming in late. I know I got yield potential. I got a couple more months. I want this crop to be green to get us to black layer.
So the only comment I would say about that R4 application is, like, for us in Wisconsin, logistics are a problem. So that's the only caution I would say. And I don't know, Marty, if you have any of those issues in Michigan, but the planes move up from Illinois here and they just keep moving north. So sometimes just trying to get planes at that R4 is a problem for us. So while we, if you can control the applications, that's fine. But that's why I think it's a little bit playing with fire, at least in my geography, because of just logistical issues, right? So that's usually why I try to get folks to kind of think about it at tassel and hopefully get it on by kind of that R3 to R4 time span. I don't know, Marty, what you see in Michigan.
Yeah, I mean, we've seen very rare instances where we spray like at the very beginning of Dent and still protect 20 bushels. Mm-hmm. I probably shouldn't even say that, right? Because someone's going to hear that and say, "I spray at Dent." Like, that's not what I'm saying. That happened once. We tried to repeat that again. That was back in 2019. We've been trying to repeat it every year since and haven't been able to see that. So that was just because disease was coming in super late. They'd already put an application on actually at tassel anyway. So just the perfect sort of storm late in the season, because the corn plant certainly does have a lot of, grain fill going on through that Dent period. But, I think Darcy's point here is really just not to let it get too bad. If you can see it driving by from the truck, you're done. It's toast. Right. So
yeah. And I guess my other comment is that early on the VTR2 timeframe, I really haven't seen them fail in our trials. But I have been in fields where they waited too long and went at R3 and then they sprayed again, and that did nothing because they waited too late, because the disease level was too severe. So that's where by 5%, if you're at 5% in that upper canopy, you missed it. Don't do anything other than if we're going to spray then we're going to probably push fungicide resistance, was a whole nother talk on that. But we got to be ahead of it. And I think you can get out and find it. And so that's why you have the maps and the apps and things to make a decision. Is it active? Are we at a stage where it's active to make a decision? That VTR2 timeframe has been pretty good for us in Indiana, even, and I know, Marty, you've had work at the R3, so that's the window we need to be in. It all depends on when the crop was planted. And I can say this year flipped the game on us. Our early planting is what got socked in Indiana, not the later planting, but we were trying to think we could escape the disease because it comes in late. But all my early plantings are where we really got the socks knocked off of us when this disease moved in this year. But again, it's watching that time frame. And then the other side of when we try to look at, does it pay to treat for tar spot? And so we actually had locations that didn't have tar spot back in those years. And so we compared that, just looking at our fungicides, can we, what is the net return compared to a low site? And this is just proof of concept where we have high disease pressure. We got a 9.5 bushel acre response. Where we had low disease pressure, we got 3 bushels, which is on standard to what we normally find across all our efficacy trials we've run across the Midwest United States. A low disease pressure, 1 to 3 bushel yield response possibly, right? That's not going to pay in today's market. And then if we looked at timing, we had a similar thing where, yes, under high disease, I think if you click it again, Damon, high disease pressure, but this is with Trivapro, we netted about 4 bushels in that high location versus -2.5 in the lower location. But if we ran the numbers one more time, Damon, on average, where we have high tar spot disease pressure, it did pay to use a fungicide. And this is looking at numbers back in 2022, 2021, we netted about $30 to $48 an acre to spray a fungicide. So again, proof of concept, a fungicide will protect and provide net return when the disease pressure gets high in the crop canopy. We may lose that fungicide cost if we don't have the disease pressure there. And then my final note on using fungicides is that coverage matters. So this is what I'm showing how a fungicide can really protect that leaf when we spray it. So you got a treated leaf versus a non-treated leaf, but coverage matters. So Damon, if you should, so here I'm showing a top-down look at the crop canopy. X marks the spot. You can kind of see that upper leaf probably captured the fungicide, and look where tar spot found its infection point. So click it one more time, Damon. And you can kind of see right there that upper leaf captured the product. Their product didn't land on that leaf below it. But you can also go out and assess, are our products working? Because I can look at the other part of the leaf there and say, yeah, that the disease is kept at bay on that leaf. There's an area that didn't get coverage. And so I was getting a lot of questions this year on, I've put a product out, but I still have tar spot in my field. And my reply is, you're never going to have 100% coverage of those leaves in the field, but you can go out and look and see where the coverage was. And so then if you click on the next, here's an example of another field from this year in Indiana. Click one more time and we'll zoom in a little bit. And if you click again, I'm going to show you can see the upper 2/3 of the canopy, we had good coverage, and that the coverage did not reach those lower leaves. And you can see, click one more time, you can see where that poor coverage is. And so this disease is really going to show us how well our coverage is in the crop canopy. And I don't know if I have a drone picture or where we had spray coverage issues. Nope. But this is one that we do see differences. And so it's important, if you're going to use some of these new technology and drones, to understand the effective boom width versus what they say it's going to do, because this disease is really going to show you that maybe it's more a 20-foot boom width or spray width of the drone versus a 30-foot spray width. Because we have many examples this year where the fields have been striped because they didn't have the good coverage. And that poor coverage doesn't mean the product's not working. It's just there's not enough product covering that crop canopy to get disease protection.
Yeah, great points, Darcy. Appreciate that perspective. So this is a nice slide because it shows us where we've changed tunes over time. So, more experience, we get to adjust what's going on. We get more evidence. And so, again, I think you've heard today, hybrid's really, really important. So spend your time there. Start trying to get ahead of it in South Dakota. I think that's my message. Really focus on those hybrids and try to get that information and lean on the seeds sales folks to try to figure out where that resistance is if you're concerned about it. Continue to assess risk, in your areas. Use those maps and apps and try to figure out what's happening around you. resist the temptation to go too soon. I think Darcy showed some nice examples there with some good data and those disease progress curves where you can go too early with a fungicide application. I think we got a lot of options with fungicides out there. As long as you have a 2 or 3-way mode of action, I think that's a really important piece. So, shop around, try to look at the efficacy guides. We have the Crop Protection Network. We have a tool there now, an interactive tool where you can actually sort those efficacy ratings and find out which things have the best efficacy out there, and then take that list to your local supplier and just see what kind of price you can get on some of those better products. And try to make that application in your favor. The app is available, so we got Tar Spotter. We'll hopefully have some more tools coming out this year as well to help with the timing of those things. And then, we heard a lot about managing irrigation from Tamra today. And I think, considering what's going on there, and we can talk maybe some more about this during the panel, I think that's going to be really important. And, we kind of crossed off the whole residue management thing. I don't think we get a lot of benefits there. So I don't know if I'd be too concerned with that. There's a lot of residue, a lot of corn in the Midwest. And so there's a lot of a residual inoculum here now. So I think, doing a lot of tillage is probably not needed. We just got to lean on those hybrids. I don't know, Marty, Darcy, anything you want to add here?
If we are going to go with a fungicide, make sure you leave a check strip. I think that's really important to understand your return on investment, and make sure you pencil it all out. Really important. Yeah, and I do it every year. It's just essential.
Yeah. Good point. Darcy, any parting words on this slide?
No, I think you covered it, and we can move on to the panel or see what other questions there are.
All right, sounds good. Just to thank a whole bunch of people. There's a lot of folks behind the scenes. Marty, Darcy, and I are just the pretty faces that get to give the talks. Some may not say I'm pretty, but whatever. But there's a lot of folks in our labs, a lot of cooperators. Some of those folks are on this call. A lot of funding agencies over the years. We've been dealing with this in our area almost a decade now. So we've had a lot of funding there from the various corn boards and, of course, the National Corn Growers Association and stuff. So we definitely need to point that out, and also the National Predictive Modeling Tool initiative there as well. So with that, I'm going to go ahead and stop the share here and hand things back over to Maddie and how you folks want to run the panel.
Yeah, thank you all so much. So I guess what we kind of envisioned for this is just having all the presenters, kind of have their cameras on, mics on, and let our attendees ask questions. I do know we have quite a few questions that have gone through Q&A, and I know that Darcy has been answering those. So I thought we could start by, finishing up what's in Q&A and chat, and then we'll just kind of go where it takes us. Does that work for everyone? All right, so I'm going to start with questions in Q&A, and we can answer them live and just kind of get going. All right, so our first question that we have in Q&A, is there a list somewhere of susceptible hybrids that we have found so far?
The $10,000 question. I guess what I would say is, due to the high, at least in my geography, due to the high hybrid turnover rates, it's been really hard to put our finger on it. You can find them. Like, I can go in our agronomist hybrid trials, and they're there, right? And we report those hybrids, and then next year some of those disappear. And so it's been really hard to sort of get a handle on that. But I think if you have access to, in your states or in surrounding states, to some of the hybrid trials that are run in those states, I know at least in Wisconsin we run, I think it's 6 or 7 locations around the state with a range of hybrids. That's good data. That's independent data, and try to get a look and then see if you can source those hybrids. I know some of the larger companies now are starting to at least they know where those hybrids rank. And so they're starting to offer some of that, at least when you go in and push them a little bit. And I hear some of the companies actually do include those ratings now in some of the catalogs. But at least that's kind of what's going on in my geography, and I'll let my colleagues kind of comment.
I'll say, unfortunately for Indiana, we no longer run hybrid trials due to lack of funding, so we don't have them as readily available. So fortunately, we have to flex on whatever's available to us, and it changes every year.
Same here, just like Damon said, you know, those hybrids change so quickly. We rely on the companies, and they often have a good idea what to expect from a hybrid, even if they don't have a rating in a catalog.
Yeah, it's similar here in Minnesota. We do run some hybrid trials still, or we have, but they have not been generally in locations where there's been much tar spots. So we don't have a lot of data from those.
One thing we didn't touch on is we did take a look at maturity group a few years back, and so and I see this especially, you know, we push maturity groups, especially in or I'm sorry, relative maturities, especially in our silage systems here, and that's where I see, you know, not only do we have some brown midribs, which are typically a little more susceptible, but we're really pushing maturity group, which keeps that plant green longer, and so we see, you know, this effect where the tar spot fungus can really continue to run on those long maturity groups. So I'm not saying to go to, you know, plant a whole bunch of short maturity groups on your farm, but be aware of that. You know, if you're pushing or you're working with a farmer who's pushing mature or relative, I keep going back to soybeans, Marty. But if you're pushing relative maturities really hard, that does leave that window open for the fungus there. So just be aware of that as well.
Mm-hmm.
Damon, along those lines, can I ask a follow-up question for you? You talked about silage. I know most of yours would probably be used for dairy. Out this way, we've got a lot of cattle on feed and feedlots, and a lot of people are chopping silage too. They've noticed rapid dry down, often making them miss the optimum window for silage. Have any of you, especially you, Damon, seen any advantage of fungicides economically on silage? I have not recommended it generally, but I've got a lot more questions recently.
Yeah, Marty and I both got data and probably some opinions on that, Tamra. You know, Marty has a really nice slide where he shows, you know, high tar spot levels in a silage field and a lower tar spot. And just, you know, my biggest concern, you know, the feed folks always look at the changes in the plant with undigestible, you know, components. And that is something, you get a lower quality feed for sure. But the even bigger one for me is the extreme drop in moisture really fast. And we actually had a trial in 2018, which was our first really bad epidemic year. It got hammered. It was a set of BMR hybrids. We actually checked it before Labor Day weekend, and we were at around, if I remember right, it was around 72% moisture, something like that. By the time we came back after the holiday on Tuesday, we checked moisture again. We were already down in the mid 50s. So it drops really fast. And my concern there is we deal a lot with mycotoxin problems. And this fungus does not lead directly to mycotoxins. Nobody's shown a toxin associated with this fungus. But what happens indirectly is we can have some of those years where we have high amounts of ear rots and some of these things. And when you start to try to make poor silage out of really too dry corn, we continue to see mycotoxins increase in the bunker. So you get all these sort of secondary issues, right? And that sort of brings me full circle around to the fungicide question. And like, you know, we've seen some inconsistencies there, but fungicides, at least in a really susceptible hybrid in a silage system, it's hard to put some economics to it. But it does help, at least, I think, slow the fungus down enough so that we can maybe get a little more flexibility out of the moisture side of things. That's kind of what I see from the silage side. So you may not notice a lot of, like, you know, strong benefits in terms of, you know, mycotoxin reductions and some of these other things, but it can help us retain some moisture in that plant so we can at least get it harvested and try to get those bunkers packed, you know. So that's kind of what I see. Marty, I don't know if you have any follow-up or opinions on that.
I think you did a really good job of covering that. We've run 3 years of trials now. We brought a student in to work on this tar spot, and then of course we didn't get any tar spot. Plenty of mycotoxins though, so Some really interesting data there looking at various products and how they suppress mycotoxins. And so I think that's where a lot of that work's going to go. But the other sort of benefit is we do have the data to start pulling out, like, is there any other benefit from the fungicide in low foliar disease years? So that'll be exciting to get that finished up.
Okay, awesome. Thank y'all. Our next question is, does anyone know if fungicides negatively affect soil fungi?
I can have a quick stab at this. So we did some work in a till-no-till system, basically demonstrating that fungicides obviously affect fungi and non-target fungi as well, including yeast. And what we actually saw in no-till, it was the microbiome was more resilient and came back quicker than in tillage system. But we saw no effect using the tools and the way we did the trial on those soil communities. I'm not saying that there can't be an effect. And I think the bigger concern is fungicide resistance, especially the triazole chemistries, those DMI chemistries, because they're used in human medicine as well. So that's probably our bigger concern, driving any resistance, not only in the plant pathogens, but those potential human pathogens too.
Okay. Anyone else have input on this one, or are we ready to move to the next question? Okay. Next question. Is a high volume ground application better than a low volume air application?
So I'll jump in because I just looked at his slides, but Darren Mueller's been actually looking at this. And then before that, Albert Tenuta up in Ontario had shown a couple of nice slide decks. And I know Darcy might have some data here on this call as well, but I can just kind of give you the cliff notes out of Darren's work because I just presented that at a conference a couple of weeks ago. And that really surprising to me, if you he was looking at drone compared to aircraft and then a ground application. So you had 15 gallons on the ground application, 2 gallons on the aircraft, and I think it was 2.5 in the drone is what they were using in terms of volume there. And really surprising, they actually got improved coverage down the plant with a drone due to the air, the push from the prop to get the spray down in there. And they've got some dye work and some other things. And I think everybody was sort of surprised just how well the aircraft and the drone actually do in terms of redistribution. I also think that we get a lot of flexibility out of our products. Aren't highly mobile in the plant, but they are more mobile than some of our older contact only fungicides. And I think that helps us a little bit with these aircraft applications. But what they were showing is they actually had really nice yield responses out of the drone, where they also controlled the overlap. And I think this is the biggest thing when you're looking at even aircraft, like we can see this as well, but it turns out the drone just is really blatant when it happens. When you don't get the overlap right, that it goes south in a hurry. But if you take some time to really calibrate that sprayer and get those overlaps right, like the drone turns out to be a really great tool, and maybe even a little bit better than a traditional ground application at 15 gallons per acre. At least that's the data I've seen. I don't know, Darcy, what your comments would be.
Well, I was just looking at some data I just saw today that changed a little bit, but also I'm going to acknowledge Craig mentioned that coverage more detail and then the nozzles. And so it comes back to, we need to understand how we're set up to begin with. So nozzle selection, we just had a class yesterday on nozzles for our grad students. It's essential to understand the coverage, and where I think things are failing is they're not understanding the actual, what is our coverage area versus the drones not working as well. But I was just looking at, so what we did is and this is one trial where we actually had some separation on tar spot disease differences, and I'm looking at it here. We actually saw the best disease reduction in the drone, but at 5 gallons per acre. So they had to slow the drone down versus the 2 gallons per acre because it's going too fast, but they slowed it down. And this is looking directly in the center of the plot where we were targeting, which, on your days, if you're targeting, and we've had these moments where just a 5-mile-an-hour wind moved the target off of what we were trying to measure. But it actually outperformed the ground rig in this trial statistically. So I had about 3% tar spot with that 5 gallons per acre drone with a T30, and then 8% in the with the ground rig, which were they were all less than the untreated of 15%. That being said, this trial really didn't have a yield of influence. So again, just all depends when the disease moved in. But I say we have this disease now that's really going to show how good the coverage is, how well is it penetrating the crop canopy, to make that assessment moving forward.
All right, great. Anyone else have anything to add, Tamra?
I think this might be a good spot to add it. We're getting a lot of questions about that on this end too. So another thing that has come up in recent months after 2024, where we saw more disease in South Central Nebraska, people are using chemigation now to apply fungicides, and we're just now starting to scratch the surface of that with more data. I'd love to hear if anybody has any data for chemigation and maybe even volumes. The second part of that is there's some very compelling, of large volume of observations from here in Nebraska this past year. I've seen a lot of imagery from up above, where a lot of times apparently people don't shut their pivots off when they have the plane come in and spray fungicides. And there's some really compelling images showing it looks very different right to the line where the pivot was still watering. And then for a few hours thereafter, as it's made its pass around, it almost gives the appearance that we've got some rainfast issues or water issues. And so I just wanted to hear some comments from some of you. I don't have data, just some really good images.
Tamara, we did some on-farm work with a grower with chemigation. So I think the trick there is to make sure you're using a very low volume of water. So let's see, I think a 7/100ths of an inch of water is what will hang in the canopy. Anything more than that, that starts to run through the canopy and off onto the ground. So we want to run those pivots quickly, putting down about 1/10 of an inch. I think most labels, run up to 1/4 of an inch, but at 1/4 an inch, you're probably losing half of the product or something down into the ground. Right. So where we saw reasonable results, we're putting down 1/10 of an inch, check uniformity of pivots, all that sort of thing. Make sure you got backflow prevention valves. My irrigation guide yell at me if I didn't mention that. But yeah, it works. We've seen some data on farm trials get suppression.
One thing I'll follow up to, the quality of your application. We know coverage is important. One thing that did go wrong one time that I want to caution everyone listening, if you're going to try chemigation, be sure to calibrate your chemigation pump with product and not with water, because sometimes these products aren't getting put on at the rate you think they are if you calibrated it with water.
Tamara, I guess we have a lot of water in Michigan, so we don't really care when we irrigate. We're pretty irresponsible, perhaps. Maybe I shouldn't say that, but most of our systems are going to be over the top, right? So if you've got drop nozzles to reduce evaporation and whatnot, that's also going to be a consideration in your situation. What's your general setup back there in Nebraska?
In Nebraska, most of our nozzles are putting on water above the canopy.
Okay.
We've just begun to look at low and mid elevation MESA and LISA drop nozzles in the canopy. And from 1 year of data, it's about what you would expect, but we had such low disease severity, I haven't been sharing it widely, but we have interest in that. This year we have planned with some of the drag lines that we can put on, and we actually hope to look at different points along the pivot too. Maybe that's a way we can mitigate disease in the inner span or two, putting water down lower in the canopy instead of over the top for so long.
Right.
But again, I don't have data yet, but it's something we plan to try.
Yeah,
Thank you.
It's interesting.
Okay, great. Our next question is, do you have any indication of plant physiology factors that correlate well to resistance? Seeing a lot of heads nodding no, and I also don't know anything. So I guess our answer is going to be no. I'm sure that's something that we will keep looking into as we go. It's important to remember that we've really only been dealing with this disease 10 years, very technically. So we're still learning a lot. Our next question is willing to give a quick summary of southern rust. I'm not sure if you're wanting just like an overview of southern rust. I know I can tell you for South Dakota, we had southern rust in at least 48 counties this last year. It did start showing up in very low amounts in late July, and it and tar spot outbreaks kind of corresponded time-wise. We started seeing severe southern rust in late August, and that's also when tar spot started showing up. We believe here in South Dakota, just because both diseases are windblown, that's the reason we were seeing them in the same counties and the same fields. And like others pointed out, we did see more southern rust in 2024 than we did tar spot, at least it's an incidence wise, and also severity wise. Does anyone else, maybe our Minnesota group or Nebraska group, want to just like briefly touch on southern rust in those states?
I can briefly comment it on Minnesota. Yes, we saw more southern rust in Minnesota in 2024 than we've probably ever seen here before. We have found it before, but never at levels or the extent that we saw this year. I'd say in a lot of fields, especially, well, in all across the southern half of the state, there are many fields where southern rust was more severe and higher incidence than tar spot. And like Tamra said before, late in the season, as the lesions, as those pustules changed to black in color, there was a lot of confusion in terms of diagnosis. So there were a lot of questions that came in about that, whether it was tar spot or rust. Some areas southern rust was pretty significant, probably at levels that result in some yield loss. But the big question is, how often will it come again, and will it be here next year, given that the pathogen doesn't survive in our region?
Very true. Tamra?
I think that's an important point, that we're still reminding people that just because you had a lot of southern rust doesn't mean you'll see it again this year, because it does not overwinter. That being said, we have to watch conditions south of us, and maybe we can put the southern rust map in the chat too to show people we do monitor that, but there's really no incentive for the southern states to populate that map. It only helps us further north, maybe not them as much. And so you may not see as many red counties down there that act and representative of what they're actually seeing down there. So that, I think that was important. And this was the worst southern rust we've seen since 2006, not long after I started. And lots of reports, a few pictures, people, windshields covered with orange. It looks like Cheeto dust when you come out of the field.
All right. Does anyone else have anything to add? Or are we ready for the next question? Okay, I'll move to our next question. Thank you all. We've kind of covered this a little bit, but so can you talk about coverage in more detail? Applying a product with a higher gallon per acre does not necessarily mean better coverage. The aircraft setup and nozzle selection is essential for optimum coverage. I feel like we've talked about this, but I didn't know if anyone else had anything they wanted to add.
No, I think we covered it. I think it's, we need to really think about what the actual spray coverage is when we're looking at changing equipment.
Yes. All right. So our next question, have you had any research with 14 ounces of Quilt Excel? If not, any comments on why or why not that may not be as good as the latest 3 mode of action products?
Yeah, this is an interesting question. I just pulled up our fungicide efficacy tool on the Crop Protection Network and sorted it, and I see that we have it listed as unknown, and when that happens, that's because we haven't had enough data to actually look at it in a standalone tar spot trial. And I think part of that reason is that product has been around for a long time, and it hasn't been one that we've been getting a lot of questions on, and so it wasn't included in some targeted tar spot trials. But there is another tool on the Crop Protection Network called the Corn Fungicide ROI Calculator that tool is actually it actually does include quiltic cell. The basis of that those sets of trials were actually run trials to try to get a look at return on investment in both low and high disease situations. And in that case, we do have a lot of tar spot data and also southern rust data in there. And so it does give some return on investment, but it's there's products that are in there that are much better now. So I think, I think where I'm going is based on what we know so far, which is limited. It's probably going to be middle of the pack in terms of those two ways. And there's probably some other things in there. And I'd encourage folks to, I'll drop the links to both of these tools in there while my colleagues talk about some of this other stuff. But I'd say it's probably one of the ones that's going to be kind of in the middle of the pack, maybe not a standout.
Yeah, Damon, and I saw the question I pulled. We had it in 2019 in some of our trials, and it looks like it's fallen in the middle of the pack for that just one specific trial that I can look at here.
All right, anything else to add from other presenters? Okay, I thought that it might be interesting to go back through a couple of the questions that Darcy was answering as we were presenting and just see if anyone else had anything else to add. So one question that was asked early on is how late is beginning of infection going to affect yield? Example, if I find it at R3, should I treat with a fungicide? And Darcy had replied, possibly if you're starting to find it mid-canopy.
Yeah.
So is anyone else? Oh, sorry, Darcy, carry on.
Yeah, so I guess my comment is if you find it at R3 and we got one lesion, then I'm not so worried about it. But if you get out there and you're finding more in the lower canopy, then I might look at that at trying to protect some of that because R3 still falls within my window of I might try to trigger something. But again, it's depending on where it is, where we're landing and how much disease you're actually seeing in the field.
Yeah, I agree with that. Allison Robertson taught me years ago with any of these corn diseases, you can use the bottom canopy as your indicator. And so, I still like that even with tar spot. When you're seeing it low in the canopy on 50% or more of the plants, and it hasn't moved up, it's go time. But if it's already up in the middle part, it's game over. And that's going to change. It's going to move around year in and year out, just, follows the weather. I mean, you've heard all that today where Darcy was commenting about how, in most years it's been late, and then last year they got burned on the early planting. So, it really just comes down to paying attention to the maps, the apps, and combining that with your scouting to truly pin down that optimal timing, in my opinion.
Okay, great. Thank you. Another one that we had was let's see, it's a very long question, so I'll read it. So great presentation, guys. I especially like the emphasis of not abandoning no-till practices and pursuing resistant hybrids. I realize that tar spot has not established itself in central and western South Dakota yet, but these regions are almost exclusively no-till and have a very established corn production enterprise. Simultaneously, there are also limited hybrid choices in these regions. If tar spot finds its way to these regions, would you suggest an emphasis on crop rotation and varying crop types? Darcy had replied, rotation as they said in their presentation, rotation's not helped much in Indiana. And when they talk rotation, it's just 1 year away with soybean. They've also seen tar spot start in fields that weren't continuous corn, but were coming off of soybean. Clarence, I don't know if you're still on. If you wanted to add anything else to that or talk, that would also be fine. Clarence is one of our extension agronomy field specialists. But anyways, does anyone else have any thoughts? I don't know exactly how long the rotations are. Typically in typically the further west we go, the longer rotations. So you're going to have corn, soybeans, wheat, maybe even sunflowers thrown in there. It's, they're 3+ year rotation cycles.
I don't think it really matters. So we've had plenty of fields here in Michigan that haven't been in corn for many years, and then they were absolutely hammered with tar spot. So yes, residue contributes to a small component, but it's so minimal I just don't think it's worth worrying about. You should be rotating, well, potentially, we would probably say, in our entomology colleagues would say you should be rotating anyway, right, for insect management and whatever else. But I think for task spot management, it's such a small component that it's, yeah.
I agree. We're the dairy state, so we got lots of alfalfa as well, and we've seen fields, out of corn for 3 or 4 years, and then they like to come in behind an alfalfa crop, and so they chose a hybrid that was susceptible, and I've seen it get pounded. So,
Yep.
so you can do all you want with a rotation, but I agree with Marty. It probably actually doesn't matter much, and I definitely don't get too wound up about residue. I mean, there's enough inoculum and enough corn in the Midwest. There's plenty of initial inoculum now that's established here. It's all about trying to really get a look at these hybrids.
So we've seen it develop in fields in Minnesota too, that had not been in corn the previous year and didn't have tar spot, at least not that we could detect 2 years before that. So clearly it is moving in from other fields, other areas. And interesting, this kind of relates to what you talked about before, Damon, in terms of the residue. We saw it on the bottom, the canopy, even though there hadn't been corn or tar spot in those fields before, as far as we know. So it makes us scratch our heads too. And I think it has to be partly the environment. That's my suspicion. We're getting better environment for earlier infection, lower, but that's my hypothesis. But again, we've seen pretty significant levels this year with no tar spot history in the fields.
I think one more addition to that question, Clarence, you mentioned central and western South Dakota. I'll second that for Nebraska and all the other Great Plains states. Our environment is much more dynamic, I guess, and changes much more the further west we go. Where for instance, I'd love to hear colleagues from the east talk about this, because the further west we travel in these Great Plains state, for about every 100 miles west, we're increasing in elevation about 1,000 feet. We're much more arid, much drier further west. Now, that'll be different in South Dakota, where you have minimal irrigation, versus Nebraska, where we do. We've got a lot of uncertainty about what to expect from tar spot as it moves further west. My personal feeling is that under pivot irrigation, we'll still see some, maybe not as bad, now that we're seeing how much change in the microclimate we're seeing. But I'd love to hear what you guys would kind of anticipate and tell growers out here in the Great Plains.
Well, I loved your irrigation slides, Tamra. The epidemiologist in me was geeking out, and
we'll share.
Yeah, no, those are great, and what it showed really nicely is, and kind of what we've been seeing, and I think also Jose's data sort of points to this, that the cooling from irrigation, I think, is what drives it. So when we ran the original models, the computers told us, and the machine learning models told us, the temperature was the only thing that we needed to worry about. And we felt a little bit, we didn't think that was quite right? So you still need to tie some biology to what the computer and the machine learning models are trying to tell us. And so we started to force various moisture parameters into those models. But what we started to learn is that the major moisture parameters that were also significant there were still tied to temperature, so i.e. relative humidity. So I like your vapor pressure deficit stuff, because that sort of helps us uncouple some of that to a certain extent. I also like dew points and dew point depressions, and we looked at dew points in some of our modeling, and those become important as well. But we ended up landing on the relative humidity surrogate for the leaf wetness. So where I'm going with all this is, like, I think even as you move more west, when you have irrigation involved, that changes that microenvironment to a point which skews it towards, and your data shows it nicely, skews it towards the microclimate that favors that, right? And so I think in those environments, you're still going to see, a conducive situation, especially in the evening hours. That seems to be a really important piece. And when you go back and even look at the mid-90s work that was done in Mexico, the Hawk paper that Jose cited, that they showed and talk about leaf wetness, especially during the nighttime hours. Right. So I think that's the really important key. Do we fall in that range? And then the other thing that we've learned, and we got burned in Wisconsin in 21, is that we were very hot. We got really hot in July, to the point where we were above 100 some days, and folks thought that was lethal, right, to the fungus, and they sort of got complacent. And when it cooled back off again, the fungus was just as happy. It slowed it down for a couple of weeks, but like high temp it just slows it. It doesn't do anything to it, right? And so when you find yourself in below average conditions and you got enough leaf wetness there, it can still move. So, unfortunately, I think, in the West, you got to keep looking for it. I think eventually it's only going to be a matter of time. And especially if you have those irrigated environments, you unfortunately have the microclimate, which I think can support it.
Thank you. I suspected that as well. It helps hearing from more experienced folks from further east.
Yeah, thank you. We do have another question in Q&A, and it says, would a general R1, R2 application, or would a R1, R2 general application be a bad idea? And I'm guessing we're asking from the context of South Dakota. I don't have any opinions on this, so I don't know if others want to kind of chime in.
I guess I'm going to draw back and tell you to look at your fields and know your field history, and then be out scouting to make a decision. I mean, if this is active early on, you're going to find it. And that's the one thing where I've heard some say you got to spray before you see, and I don't agree with that, because when we find it, I have lots of time to find it before I need to trigger. I have a good month and a half there, probably some years, because we can still find it at that V8 stage. And if we still trigger it at the R1, we still getting good protection. So that's where I'm going to say, I know you don't want the extra work and you want a one size fits all answer, but I would watch the maps and scout and be out monitoring, and then you can make that decision if you want to pull the trigger.
Yeah, what's interesting, Darcy, and your microscopy slides show this really nicely, is it takes so much time from the infection process to when you actually get that stromatal start, and your data. If you sort of took what you knew about in the photomicrographs and then you overlaid it on your disease progress curves, I mean, you can see you have some flexibility there. So it's not like you see the first stromata in the lower canopy and the crop is done, right? You actually have some flexibility there, because the fungus moves very slow in that initial phase, right? And you could see it in those disease progress curves. Yes, once you get into that really steep part of the curve where it's already into the mid canopy and it's super heavy, like that's game over. But like, it's not time to panic when you see the first stromata. So I think, the beauty of this is that you can use scouting, right? And you can make an informed decision on this fungicide application, and we can try to slow down fungicide resistance development, which is going to happen inevitably if we keep just blanket spraying everything.
Awesome. One of the things that, we had somebody in the chat. They are having issues with downloading the Tar Spotter app from Google.
Yeah, that's it. I forgot to mention that. So yeah, the Google supported version has been kicked out of the store because we didn't update it. The iPhone version will still run. We are going to be putting on the Crop Protection Network will be a new tool. It'll be a dynamic online app, which will work. One of the issues we've run into with apps over the years is that you have to build an app for the Android version, and then you have to build an app for the iPhone version, and you have to have two programmers and all these sorts of things. And the world is moving towards online dynamic apps, and so that's where we're headed. So continue to stay posted on the Crop Protection Network, and we'll have a tool there. We have a beta version. I can promise you that tool is in existence. I was actually just looking at it this morning, so it does exist. We're just working out some bugs and things, and it'll be deployed this growing season.
Awesome. Is Field Profit still available on Google, or is it also off?
Oh, I love these questions. So if anybody loved us talk about how to start a startup company and then how to kill a startup company, I'd be happy to talk with you about that. Unfortunately, Field Profit no longer exists. My co-founders, we unfortunately went out of business. My co-founders kept the IP for that tool, and so it no longer exists. So unfortunately, not available there either, but we will have everything publicly available on the Crop Protection Network.
Okay, awesome. We did get another question in. In your experience, how many acres a day can a drone treat? I think that's probably going to be a Darcy question, maybe.
I don't have the answer to that one.
Okay.
I think it's a couple of hundred, just chatting with folks here. I mean, yeah, a couple of hundred. We have guys that spraying, thousands of acres. But I don't have all the numbers and figures and whatnot, but at least a couple of hundred.
Okay, awesome. So one of the things that Connie had brought up is wondering if we wanted to pull up the ROI calculator and maybe like show how that works, kind of as a quick little demonstration. I don't know if maybe Damon or you wanted to maybe pull that up and share it on your screen. Would that be something you would want to do? Okay.
Yeah, happy to do it.
Thank you.
Let me cue it up here. Everybody see that okay?
Yeah.
Yep. See some heads nodding. Yeah, so that link, when you land on that link, you'll come to this page. Be patient. It does take a few seconds for that to load, and that could be extended just a little bit if you're in a rural internet zone. Just be patient. There's some complicated simulations that actually run in the background, and that's why it loads. So it actually has to rerun those simulations when you load it on the page, and that's all done on a remote server that's actually here at the University of Wisconsin. But what's nice about this is we have data from across the entire Corn Belt. So stuff from all the way down in Mississippi, all the way on up into Ontario, Canada is included here. And there's I forget how many site years, but there's a ton of site years of data. This is all research based. It's all university stuff. So I want you to be aware that that data set that underlies these simulations is indeed an independent, non-biased data set here. And what we did is we took all that information in terms of yield and median costs of products and things over the years that study was done, and we built it into these Monte Carlo simulations, which run in the background. And so what you can do is you can actually come in and you can use these toggles here to give yourself a low or high disease situation, so you can actually play around a little bit here. You can even click this little box here, and you could actually input your own sort of theoretical percentage if you really wanted to. I don't think you need to go to that level, but if you just want to mess around with it, it's kind of cool. What I would do though is be pretty honest with yourself and look at your field histories and your expected yields, and you can change this box. So we're defaulted right now at 180. I could move this up. We got some fields here that are gonna go to 200, and then you can also put your expected commodity price in here as well. So you can we'll leave it at 5 because the tool's doing some work right now. So you're seeing it right now. It's going back. It's running those simulations, but you can actually tailor it to your operation. So spend some time doing that. We do pre-populate the treatment costs here actually with what we knew from the time that this study was run, what the median costs were. And we actually surveyed chem suppliers to come up with those median costs. And we've taken a lot of flack from the companies over these prices, but that's what they were. And of course, nobody wants to tell us what they're actually, paying for fungicide products. So we do allow you to be able to change those costs. So you can click on those boxes and you can repopulate those with whatever your local costs are. And then you saw how the tool will actually, change in real time for you. And what you're getting here in the visualizations now is you're seeing a graph, a set of bar charts here with the expected net benefit per acre. Here's the $0 line right here in the middle, and then the gray bars just kind of show you the spread of data around the mean, which is the black bar here. I actually like the table a little more up here. It's a little more informative for me. But what this is doing is that it's giving you the same expected net benefits. And then it's also providing you the break-even probabilities. So the break-even probability is the number that we get, or the probability that we'll at least recover the cost of the fungicide. And I would say look at that number first for a lot of these products, because you can have high expected net return. In fact, Miravis Neo is a decent example here. You can have a high net benefit per acre, but your break-even probability might be actually a little bit on the lower side. And a lot of that has to relate to the cost of the product. So again, being honest with yourself and putting that cost in that you're getting locally will help adjust those break-even probabilities, and that will adjust that expected net benefit for you. And so you can see some other examples here. Quilt Excel, that came up, that's in here. The expect net benefit here 590, but it's got a high break-even probability, right? So you're likely going to at least get the cost of that product back, and that's because it's fairly cheap. So while it might not be the most efficacious, your probability of getting a high return there is actually pretty high. So we put this tool together, and I can tell you that this tool was developed in a time where we had a lot of tar spot and then also southern rust from the southern states. So when you click on those high values, that's really incorporating high tar spot years into the data set. So with that, I'll stop a screen share, and I can take any follow-up questions if anybody has anything on that tool.
Not seeing any follow-up questions just yet. I think we have finally caught up with all of the questions so far. If anyone thinks of anything else, feel free to ask it in the chat or in the Q&A. I don't know. I was trying to think. Sierra, is there anything that we have seen in South Dakota that we think it would be good to have explained by others? Nope. Yeah, I mean, you've pretty much answered most of the questions that we've been getting. One of the things that we've talked about a bit is really making sure that there's a reason to be spraying for tar spot, and then kind of like Darcy said, not spraying too late so that we don't end up having fungicide resistant tar spot. I don't know if y'all have any opinions on that, but that's one of the things that we've talked about a little bit, is like making sure that we're making good spray decisions so we don't have resistance before we actually need fungicides for control.
Darcy, you got any comments? I'm going too late.
Well, I guess that's the biggest question I'm getting in Indiana, but we're not. I mean, we are already treating probably most of the state. We're going to trigger one time moving forward after this year, but we are seeing that there's multiple applications going out. And so that's a lot of the questions I've been handling this year. And again, these R5s that I've seen don't affect, like, in addition to trying to control tar spot, they're not going to help with standability and other things. And we've gone back and pulled data. And I actually put a trial, like after hearing the guys were going to go spraying at R5, we threw again some out at R5, and I didn't get any net benefit. The disease was already moved in. We actually might have seen more lodging with the product, but it didn't separate. We need to be ahead of the game if we're going to make a decision on controlling whatever disease it is earlier in the season. And then if we just missed it, we're just going to have to note that it's there and could be there for other years. There are seasons where the disease didn't move into R5, really not affecting yield at that point. But note that it could be there now. The inoculum load is there for down the road when we finally have optimum conditions that really kick it up early on. So again, it just is a reminder, just we can reiterate, use these tools when we turn on the map in Indiana, which this year I turned it on June 10th following a Kansas report. It started early and then it dropped off. But I did have areas in southern Indiana that were starting to report early, and the disease really knocked the socks off of some of those areas where it got in early. But again, it's scouting and knowing what's going on in your farm to make a decision whether you need to pull the trigger, yes or no, because I think that's what you're trying to decide up in South Dakota is yes or no. So I would just make sure, be out scouting, seeing if it's active, and then, make that informed decision that yes, a fungicide would be necessary this year.
Yeah, the R5 thing, a couple of years ago, I was at a conference, and really great farmer. I really enjoy him, and he works with a consultant that's pretty progressive, and they just didn't believe me. They're like, no, we're gonna try some R5s. So they actually did put some strips out. They put I give them a lot of credit. They put replicated R5 strips and compared it against some tassel applications, and he came up to me in a meeting after that season and said, "Yep, you were right. R5s don't work." He basically admitted that he blew some money, putting those applications out. So, Darcy's data just really shows that it's a pretty clean break once you get to that R5 growth stage, and when it's out of hand, it really is too late. And you're putting a lot of selection pressure on all these organisms that are out there. Remember, there's a lot of other fungi out in the landscape, and so you're sort of waiting. Not only are you wasting money, but you're really pushing fungicide resistance.
And I guess I forgot I could flip it on the other side. If we're pushing the envelope on spraying later, that may also delay some of our dry down. And we're starting to pool that data to show, as we spray later, where we might increase the moisture of the corn, maybe not this last season where we dry down good, but then when we're a delayed year where it's a wet September, October, it may delay the dry down to get to where we want to harvest it as well. So some other things to think about.
Another thing I think that has come up here that's sort of related, but on the other end, we still have a lot of people that want to spray early and they feel like they're getting ahead of it doing that. I'm talking about, like the V6 applications. And one of the things I've been telling them is a lot of people aren't aware that on some of our products labels, it restricts you to only 2 applications during a growing season. I don't think most people need a second application either for tar spot here, but I would sure rather have the option if I needed it on the back end than putting it on the front end when I don't tend to see disease. So that's come up here, and I just think people don't know that some of those do have those label restrictions.
Yep, and they also have the other pre-harvest date label in as well. Some of them you got to watch that as well. And I guess I would say even with the 2-pass program where it's been effective, it's the late vegetative time frame or early tassel that we're hitting it and then coming back again. And that's not every year. We only have a few trials where that was effective. And it was just that 1 year where it started early. This year, the other side of it is I had comments come back, my 2-pass program was the one that paid, my single pass didn't. And the reality is the single pass was the earlier, it was not the time frame that was the best, it was the second pass that was the better time frame. So again, it's monitoring when the disease was active.
Okay, awesome. Well, I don't know. I don't have any more questions. Does anyone have anything else that they want to add or talk about? Anything? Otherwise, we can get done just a little bit early. Okay. Well, I guess we have talked tar spot from all the angles and from 7 different states. So we just want to say thank you all for attending today. And I also want to thank all of our presenters. This took us 5 months to get together and get everyone's schedules on the same page. And I thank you all for taking the time and sharing all of your knowledge and skill, and hopefully we will be able to, be on top of tar spot a little bit better here in South Dakota, because we are watching it and very closely and, seeing what's happening. We did have another Q&A come in. Great presentation. Thank you all for doing this. Anyways, otherwise, feel free if you're here in South Dakota to reach out to me or Connie Strunk with additional questions. And that's everything I have. Thank you all again.